Okay, I think we're good. Okay, so we'll start off with questions from you guys with digestive and reproductive. Anything that anyone would like me to start off with right off the bat? All right, awesome. You guys are all set for the exam. I love it. You don't need my help, right? Any thoughts? Should we just do it like I planned? We'll go with that. Okay, let's do that. Okay, so we'll start off with the big general overview of digestive physiology. And again, I'm going to focus on the macronutrients. Okay, so we'll start off with carbohydrates, what exactly they are, how they get digested, what secretions are involved, how they get absorbed, and what happens once they are absorbed into the blood. Okay, so keep in mind, carbohydrates are separated into two major categories, right? You have your simple carbohydrates and you have your complex carbohydrates. Okay, so simple carbohydrates are going to be your monosaccharides and disaccharides. And again, don't memorize small stuff like that, right? Look at the name. It says monosaccharide. That means it has to have one of something. It's referring to the number of sugar units that we're talking about here, right? So these you have to memorize, right? The names of the individual monosaccharides, glucose, galactose, and fructose. But the actual concept of monosaccharides or disaccharides, it's right in the name, 2 sugar units. Keep in mind, what of these are actually absorbable? By the way, I'm going to be asking a lot of questions. Just make sure no one's falling asleep. Yeah, I heard someone say it. What is it? Monosaccharides, right? So we can only absorb glucose, galactose, and fructose. That means disaccharides have to be digested down. They have to be broken apart into their individual sugar units. Same is true with the complex carbohydrates, right? These are branched polymers, as we call them, of a bunch of sugar units, right? So in this case, you can see these are literally a bunch of glucose molecules that are physically attached to each other, right? So common ones are things like starch and glycogen. Starch being the plant storage of glucose, glycogen being the animal storage of glucose. Right, so same idea. You cannot absorb these. You have to break these down into the monosaccharides. Right, so in this case, if these are all glucose molecules, you have to break them down into their individual glucose units. Okay, so let's start off with what's happening back in the mouth. Is there any chemical digestion of carbohydrates taking place in the mouth? Salivary amylase, absolutely. Now, quick question for you. Is amylase going to be digesting lactase, sucrase, or maltase? No, right? So if we go back, you see the word amylose is here next to starch. This also applies to glycogen, by the way, for all intents and purposes anyways, right? So amylase is going to break down amylose. It's not going to break down your disaccharides, right? So when it comes to what's happening in the mouth, it's the breakdown of these larger complex carbohydrates that's taking place, not the disaccharides, right? So think about what you're eating in your diet. If you're drinking milk or you're eating cheese, which contains lactose, you're not breaking down any of that in the mouth, okay? And same with sucrose. That's sugar, basically, table sugar, right? If you spritz some sugar on, I don't know, cereal or eating a donut or something, who knows? Amylase is not going to be breaking down sucrose, okay? That just, amylase does not do that, at least it doesn't do that at all, I should say. Okay, so examples of carbohydrates. You have bread and sweet potatoes, as Dr. Woods put it, right? So amylase is the enzyme that's going to be breaking down the complex carbohydrates, not the disaccharides. Okay, those are going to be coming a lot later on. What exactly does amylase do? Well, it's going to be breaking down those complex carbs, those glucose polymers, right? Those several units of glucose molecules into smaller and smaller subunits. Now, where exactly is salivary amylase coming from? Because we talked about the fact that we have 3 pairs of salivary glands. So are all 3 of those pairs of salivary glands contributing to salivary amylase? No. So does anyone remember which specific salivary glands are involved with secreting this enzyme? And it's okay if you don't know, by the way. You still have 4 days to study. I fully recognize like it's still early on, relatively speaking. Just keep up is the point. And if you do have the answer, that's great. And if you don't, feel free to note it down for something to study later on. Any thoughts? Yep. Is it the parotid? Parotid, absolutely. So you have 1 pair of salivary glands right next to the, right underneath the ear, basically. So I'll just quickly go through this. right? So saliva contains a couple things, right? Like water, ions, mucus, proteins. Enzymes is what we care about, especially for digestion, right? But here's the thing. Not all of your salivary glands are secreting enzymes, right? So when it comes to chemical digestion of the carbohydrates in your mouth, it's the parotid glands that play the major contributor, right? They're a very high volume of watery saliva and enzymes. And hopefully it makes sense that it's watery, right? Because if you're eating food, you do not want to have a dry, sticky mouth. You want to have water to be able to break it down and make it easier to chew and swallow, right? There's another pair of salivary glands that are going to be contributing to enzyme secretion, the submandibular gland. Okay, so that's located just below the mandible or the jawbone, basically. Okay, so these two glands are contributing enzyme secretions. The parotid gland is the major contributor. Submandibular gland has a little bit of enzymes, but it's really the parotid gland that's the major contributor to enzyme secretion. And keep in mind, we have a third pair of salivary gland, right? That's the sublingual gland, the one underneath the tongue. This does not contribute to digestion, right? This is just what's being made while you guys are sitting here not eating, right? It's just to help keep your mouth lubricated. No enzymes, not a lot of water, just something enough to keep your mouth lubricated and not dry, basically, to help protect your teeth. Does that make sense so far? Okay. Okay, so again, amylase is going to break down amylose. Okay, that's the nice thing with the names of these enzymes. They're usually related to what they break down. Okay, so amylase will specifically break down the polymers of glucose, all those attached sugar units of glucose. Now, again, it doesn't break them down to monosaccharides. It's going to break them down into this disaccharide and this trisaccharide here, which you don't really need to know. The point is, it's going to generate maltose. And again, keep in mind, maltose is not absorbable, right? We can only absorb monosaccharides. Okay, so that's the chemical digestion taking place in the mouth. Keep in mind, you also have mechanical digestion to literally physically break down the food to help with the chemical digestion. Is there any absorption taking place in the mouth, in the oral cavity? So most people are shaking their heads. There's a very, very tiny amount, technically. Okay, so for the most part, very little to no absorption, except for like pure glucose, which you're very rarely eating in a normal meal, and some sorts of drugs, right? The kinds that you put underneath your tongue and that can get absorbed into the body. But for the most part, you have very little to no absorption taking place in the oral cavity. Anything happening in the stomach? Yes, what's happening in the stomach? With carbohydrate digestion. Anyone else want to help out? Yep. Yeah, so actually there's no carbohydrate digestion taking place, right? So even though we have amylase in the mouth, we can swallow it, but the thing is, it actually gets inactivated in the stomach. Why is it getting inactivated in the stomach? What is exactly causing that? Go ahead. Exactly, right? So keep in mind, you might have learned this in high school bio. Enzymes are proteins, right? And proteins like to act in a certain pH. They prefer certain environments, right? So if your amylase is working well in your mouth, do you have a lot of acid in your mouth normally? No, that means it's working best in a non-acidic environment, right? So the second you swallow it, and now it's exposed to the stomach acid within the stomach, it's going to get inactivated by that stomach acid, right? And keep in mind, this is also true in reverse for the enzymes that do work well in the stomach, in an acidic environment, right? So gastric pepsin or lipase, gastric lipase, those enzymes work in the stomach, meaning they work well in a low pH. That means they don't work well in a non-low pH, right? If you're in an alkaline environment or a neutral environment, they're not going to work as well. So keep those things in mind. These are all proteins. They work in a very specific environment, right? So even though you swallow salivary amylase, it does not play any role in the stomach because that stomach acid is literally inactivating it. Does that make sense so far? Okay. What about the small intestine? A lot happening in the small intestine. What's happening specifically with carbohydrate digestion? Let's start off with complex carbs again, right? Because we started to break down the complex carbs in the mouth with that salivary amylase. But again, we're not really keeping the food in our mouth for that long. So it's possible that you could still have a lot of amylase, right? Glycogen or starch that does make it down to the small intestine. So is there another enzyme that's going to help to break down those complex carbs again within the small intestine? Hint, it's the same name as what we already talked about. Go ahead. Thank you. Pancreatic amylase, right? So keep in mind, this is not made by the small intestine. This is secreted by the pancreas, right? So what it's going to do is it's going to continue to break down the complex carbs, right? So any starch or glycogen that has made it this far can still get broken down by pancreatic amylase. And again, it's not making monosaccharides, right? It's breaking down. If we go back to that original diagram I showed you before, this diagram here, it's breaking down that amylase into basically maltose, right? These are still not absorbable units. So there has to be some additional processing that's taking place. But keep in mind, this is what we do to finish off the complex carbohydrate digestion. Where is this pancreatic amylase coming from specifically? Obviously the pancreas, but there's a couple different cell types within the pancreas, right? So does anyone remember the specific name of the pancreatic cells that are secreting this enzyme? It's a multiple choice exam, so I'll give you options. Is it ductal cells, beta cells, alpha cells, acinar cells? Hint, hint. It's acinar cells. And the answer is acinar cells, right? So the acinar cells, keep in mind, this is like what the pancreas looks like underneath the microscope, right? You have a bunch of clusters of cells. These are your acinar cells, the orange ones. You have your endocrine cells, which are making things like insulin, glucagon, and somatostatin, which I don't think we actually talked about this year. And you also have ductal cells, right? So the cells that are literally making up the ducts, the pancreatic ducts of the pancreas. The acinar cells, these cluster of like these orangish pinkish cells, they're the ones that are making your exocrine secretions, right? So things that are being put into the small intestine. Okay, so specifically when it comes to carbohydrate digestion, you have pancreatic amylase. Again, it's going to break down the complex carbs that have made it this far into the small intestine. Okay, so again, breaking down amylase into maltose and technically maltotriose as well, which again, I don't think you have to worry too much about here. But now I'll point your attention to this. We have a lot of maltose now, right? That's been broken down by this amylase. You may have ingested some lactose, especially if you've eaten some dairy product like cheese, for example. And you might have eaten some sucrose if you had a donut or, I don't know, some sugary food or whatever. Again, amylase is not acting on any of these. But can we absorb any of these disaccharides down here? We have to break them down still. We have to break them down to the monosaccharides because that is what our body actually can absorb. These are way too big. So what do we rely on now to break up these disaccharides? I know you have the answer. See if anyone else can contribute here. Yep. Absolutely, right? So you have your brush border enzymes. These are found specifically in your small intestine now, right? So these are not pancreatic enzymes. These are physically attached, found in the small intestine part of the membrane of the cells. Okay, so what are the brush border enzymes? You have lactase, sucrase, and maltase. And that's pretty handy, right? They're named for exactly what they're involved in breaking down. Lactase is going to break down lactose. Sucrase breaks down sucrose. Maltase breaks down maltose. Okay, these, unfortunately, you kind of have to memorize, like the specific monosaccharides that come from each of these. I'll give you a hint, though. Galactose is going to come from lactose. All of them have glucose, and fructose sounds pretty sweet, so that's why I kind of attach it to sucrase, or sucrose, rather. Okay, but yeah, lactose gets broken down into glucose and galactose, sucrose into glucose and fructose, and then maltose into 2 glucose monosaccharides. Right, so again, keep in mind, where are these enzymes? They're literally a part of these small intestinal cells, right? So remember, these are your 4 layers of your small intestine, right? You have your mucosal layer, submucosal layer, the muscle layer, and then the serosa layer. Those brush border enzymes are found up here in the mucosal layer because this is what's facing the inside of the small intestine, right? So keep in mind, remember, the mucosal layer has all of these villi, right? Those finger-like projections sticking out into the inside of the small intestine. And then when you look at those villi, if you look at the cells that make up the villi, they have these little like brushes on them, right? Those are the brush borders, right? That's why we refer to it as the brush border. And the enzymes are physically part of that brush border, which is why we call them brush border enzymes. Okay, so lactase, sucrase, and maltase. These are finally going to break down the disaccharides into lactose, sucrose, and maltose. And then look at that. We have a bunch of monosaccharides now. We're good, right? We can absorb these, right? And that's exactly what's going to happen. Right, so glucose and galactose can simply move into the cell with a sodium symporter, right? So hopefully this transporter sounds familiar to everyone, right? As you're starting to make connections and studying through all the material, make connections on other material within the course. You've seen this transporter before, right? The sodium glucose symporter you've seen in the proximal tubule, right? Remember in the kidney, this is the one that's going to be reabsorbing all the filtered glucose that has made it into the lumen of the tubule, right? So this is going to bring glucose and galactose, because these two are very similar in their shape, into the cell with sodium, right? So regardless of what the concentration gradient is of sodium of glucose and galactose, we know that sodium is low inside of cells, right? So the fact that there's such a high gradient means that as sodium is being pulled in, it's going to literally drag all the glucose and galactose with it. This is a way that you can maximize glucose and galactose absorption. Does that make sense so far? Do you have any questions? Are we doing okay? How's the pacing? Is it too fast? Okay, awesome. Keep in mind, we still have one other monosaccharide here, right? So we also have fructose. So this is a little bit more simpler. We just have a uniporter here, which is just going to bring it down its concentration gradient into the absorptive cells. By the way, where are these absorptive cells? Where's this all happening? Sorry, say that again? Perfect, exactly, right? So this is all in the small intestine, and I love that. You said the apical membrane, right? So keep those terminologies in mind, right? It's this exact same things that you guys learned about in the kidney section. The apical or luminal membrane is what's facing the inside of the small intestine, the lumen, versus the basolateral membrane on the other side is facing the other side, basically, right? So same with the kidney cells and the tubule cells, right? It's the same idea there. Now you have your monosaccharides in the absorptive cells. By the way, why do we need transporters? Why can't these just go across the membrane? Go ahead. Exactly, right? So now I'm tying in what we learned about in the very first unit, right? Excitable cell unit. Things that can pass the cell membrane are small and hydrophobic or lipophilic. These are way too big, and they are hydrophilic. Okay, so they literally cannot pass through the membrane because, again, you have the phospholipid membrane, right? The hydrophobic tails that act as a barrier to prevent them from moving through, right? So they have to have a transporter to allow them to move through. But it's no good if fructose, glucose, and galactose are just staying in these cells, right? You want to get it into the blood so that the body can use it, right? You need the cells of your body to have access to nutrients. So then you have another uniporter, a monosaccharide uniporter, that can basically move these monosaccharides into the blood. And now it's in the body. It can get distributed by the circulatory system. Your heart's going to pump away, and all of your cells now have access to those monosaccharides. Okay, and keep in mind, pretty much every single cell in our body has this right over here, your sodium potassium ATPase, right? So going back to unit number 1, this is going to keep sodium nice and low in the cells, right? It's pumping out 3 sodiums and bringing in 2 potassiums, right? That's against their gradients, right? You have way more sodium outside of cells and way more potassium inside of cells. Okay, remember salty banana? So this pump needs ATP to physically move these things against their membrane. And this is what keeps sodium nice and low in the cell and allows this transporter to keep working. Is it all good so far? Okay. Now let's keep going with what happens to those carbohydrates now that they're absorbed into the blood. Keep in mind, they're not going to actually get distributed throughout the whole body right away, right? The first thing that happens is that they're actually going to travel to the liver, right? So remember, you have that hepatic portal vein, right? That is the blood vessel that's taking nutrient-rich blood, as we called it, right? So we're literally taking blood that's coming from the intestines and taking it to the liver. And that's going to combine with your oxygen-rich blood from your hepatic artery, right? Those come together in those small vessels called sinusoids, if that rings a bell to anyone, right? And the liver is going to basically convert all of those monosaccharides into glucose if they're not already glucose, right? Why do this? Because our body prefers glucose, right? Think of glycolysis and all those steps that you guys learned about in high school bio. Our body prefers glucose. That is the main source of nutrient that our cells use to burn fuel and make energy, right? So fructose and galactose are all going to get converted into glucose in the liver. And if necessary, the liver can make other decisions too, right? If, for example, we have tons and tons of nutrients in our body, the liver can just store it as glycogen then, right? Just, it's trying to be efficient with the process. The liver is what makes those sort of decisions. Okay, so that's great. Now the liver has converted everything into glucose. Then what? Now it's going to get pumped to the heart, and then the heart's going to pump it to the rest of the body. What happens next? What is glucose used for in the body? What do we need glucose for? Why do we eat at all? I hate food, by the way. Did I ever tell anyone that? I hate the idea of food. Like, I'd love to have just a pill that gives you all the calories and nutrients you need and not waste time eating and cooking food and all that nonsense. Anyways, I know I'm in the minority by saying that. The point is, what do we need food for? I heard someone say it. Yeah, exactly, right? The whole point of nutrients is for our cells to make energy, right? So here I have your carbohydrate fates. Number one, ATP production, right? So glucose is literally going to be taken up by your cells and it's going to be used to make energy, right? So it's going to go through all those steps, glycolysis, the citric acid cycle, the electron transport system, which we'll talk about here in a second, ultimately to make energy. Why do we need energy? Well, so our cells can function, right? Like, sodium, potassium, ATPases need energy. The production of proteins needs energy. Your heart beating needs energy. Everything needs energy, right? So this is what's used to allow us to live. Me moving my arms around, I'm contracting myosin and actin here right now, right? The whole sliding filament theory, all of that needs energy, right? So you have to have glucose being distributed to all the parts of your body for this. Here's a really cool other function, though. If you have a very protein-poor diet and you're not ingesting enough proteins, your body can also convert glucose into some amino acids, right? So if, for instance, you don't have enough actin and myosin and you're lacking the amino acids to make that, you can literally make the amino acids that are contributing to those proteins by converting glucose into them. Now, keep in mind, this doesn't happen with all amino acids. Certain amino acids can do this, though. Okay, then what? So now your cells are happy. They have enough energy to survive. They're carrying out their functions, and they have all the amino acids they need. What's the next step? Do you think the body wants to waste glucose? No. So what's the next step now? And the answer's on the slide already, but I'll just say it. It's to make storage forms, right? It's to basically store the remaining nutrients. Again, keep in mind, we've evolved from caveman days. We did not have access to Walmarts and McDonald's back in those times, right? Food was not just plentiful and easily accessed. Our bodies want to be as efficient as possible at storing nutrients because you don't know when the next meal is coming, right? You don't know when the next animal or the next, berry in the wild you're gonna come across that, right? So the body is very, very efficient at storing the excess nutrients. You do not want to waste it. Keep this in mind, right? Remember the renal section? We talked about the sodium glucose symporter. That was supposed to absorb, or rather reabsorb, all the glucose molecules, right? Same idea. You do not want to waste glucose. You don't want to urinate it away. That's a bad use of that nutrient, right? So first thing that happens, you're going to store it in short-term storage, glycogen, right? So glycogen is basically, remember we talked about it already. It's the branched form of glucose. Right, it was the amylose basically, right? The attached units, the several units of glucose molecules attached to each other. But here's the thing: we have a limited capacity to store glycogen, right? So what happens once you've filled up those capacities and you still have glucose remaining? What's the next step? Well, the next step then is long-term storage, right? Fats. You store them as triglycerides specifically, right? So if you eat a very high carb diet, you're going to put on weight because guess what? You're going to have enough to do all these first three things. And then the excess, the rest of it is going to be stored in fat, right? It's going to be stored for later when you're not eating food. Any questions on any of this so far? Are we still doing okay? Okay. And again, if this is information overload, don't worry too much about it. You still have 4 days to study. As long as you can keep up and kind of like follow the logic, that's the main thing, right? You can spend the next 4 days trying to remember all the specific minute details. The point is, I want to make sure you guys understand the material. It's way easier to remember things if you understand them rather than memorize them. Let's get into a couple more details here, specifically starting off with this ATP production, right? So if we go back to this liver part, this previous slide, we talked about the fact that now the glucose is in the blood and the heart is basically going to be pumping that blood with the glucose in it to all parts of the body. How exactly does glucose get into the cells to be used to make energy? Does anyone remember the specific hormone that the pancreas makes that does this basically to lower blood glucose levels? Yeah, I heard it. What was it? Come on, have confidence. What do people with diabetes lack? Insulin, right? So remember, insulin is the hormone made by the pancreas, specifically the beta cells. They're going to release that insulin into the blood. And what it does is it basically stimulates glucose uptake, right? So glucose is in the blood right now, but you don't want the glucose to be in the blood. What good is that? You want it to get into the cells, right? So you have the cells of your body, right? This is all cells of your body, liver cells, heart cells, kidney cells, the cells of your brain, the cells of your pinky toe. They're going to take up that glucose from the blood into them to make energy. How does it do that? Remember, glucose is a large molecule and a hydrophilic molecule. It can't pass through the cell membrane. It requires a transporter for it to get into the cell. Right? So we have glucose uniporters present in the membrane to allow the uptake of glucose from the blood into the cell. And because it's a uniporter, it's moving glucose down a concentration gradient. Right? So after you've eaten a meal, presumably you're going to have relatively high glucose in your blood. Right? So that's fine. That glucose can just go from a high concentration from your blood into the cells then, and then they can be used to make energy. Any questions on that so far? And then this is what happens. Oh, that does not look proper on that slide. But anyways, this is what happens for the purposes of making energy. Now, Dr. Woods is pretty straightforward about this and not having to memorize or know all the specific details. The 4 key steps that you do have to remember. Glycolysis makes a little bit of ATP. You're basically breaking down glucose. Okay, that's what glycolysis means, lysis of glucose. Okay, so that's going to break glucose, and you're going to generate this molecule, pyruvic acid. Okay, a kind of consequence of this is you make a little bit of energy. Now, I don't remember if Dr. Woods mentioned the specific numbers, but I believe it's 2 molecules of ATP. It's like a really, really, really tiny amount, but a small amount that still gets made. That pyruvic acid, though, then goes on. This is now being put into the mitochondria of the cells to make then this molecule, acetyl coenzyme A. What does that do? Well, it's going to be put in something called the citric acid cycle. This is what's making your carbon dioxide, by the way, right? So keep in mind, now we're thinking about respiratory units here, right? Where does that carbon dioxide come from? Where does the waste product come from? It's coming from this right here, your citric acid cycle. This is the waste product that's being made as your cells are working away. This is why we have to exhale carbon dioxide to get rid of that waste product. Okay, and then another thing that happens is you make, again, a very small amount of ATP. Again, I think it's like 2 molecules. But then you also make these things over here, hydrogen ions and high energy electron carriers. You don't need to know what those are. What you do need to know is that they can enter into your electron transport chain, and that is going to make a crap load of ATP. This is where the vast majority of energy is coming from right over here. Okay, so keep in mind, all of this is happening within the mitochondria of the cell. What did you guys learn about the mitochondria from high school bio? It's the powerhouse of the cell, right? This is why it's making all of that ATP. A little bit over here, a ton over here in the electron transport system. That is all you have to know about this. In reality, there's like 10 steps over here. God knows how many steps over here. That's all second-year biochem that I forgot years ago. Study well. Don't be me. Don't memorize things and forget them later on. Any questions on any of this so far? Are we still okay? Keeping up? Anyone need a break to like cry or anything? Can I go cry? Okay. Okay, so keep in mind, that's all happening in your fed state, right? The production of ATP. What happens after you've made enough energy and you presumably have enough amino acids in your body? Well, then you start to go with short-term storage, right? Again, you do not want to waste the extra glucose. You need that glucose to survive. So first thing that happens is that you're going to make glycogen, right? That is the storage form of glucose. And by the way, look at the kind of structure of glycogen here, right? It's literally a bunch of glucose molecules glued together and kind of like this branch structure as well, right? So that's how you get that amylose idea. This is what amylase is going to be breaking down. Now, a lot of cells can store glycogen, but the 2 main types of tissues that do it the best are the skeletal muscle and the liver. Okay, they have the enzymes and they have the physical capacity to store glycogen the best. Okay, so these are the 2 main tissues for storing glycogen. There are tissues that are completely incapable of storing glycogen, right? And it's on the slide here. It's the brain and your nervous tissue, right? So all your neurons of your peripheral nervous system and the brain itself cannot store glycogen. And this is a really, really important point, right? Because keep in mind, your brain is a very metabolically active tissue. Think about all the action potentials and everything we talked about. Think about all the energy that's needed to allow those to happen, right? So the fact that they can't store glycogen is really important because they need glucose constantly, right? For you to think, for you to move around, for you to do anything. All those action potentials constantly need energy and therefore they need glucose, right? So there has to be some sort of other mechanism in place to make sure that your nervous tissue still has access to those glucose molecules, which we'll get into here in just a second. Okay, so this is somewhat important. Glucose being converted to glucose 6-phosphate. This addition of the phosphate group, by the way, it's called this because you put the phosphate on the 6th carbon of glucose. That's how you get the name glucose 6-phosphate. I don't think you need to know those specific details, but this is actually, by the way, the first step of glycolysis, right? So in theory, you could take this and go on to glycolysis, form your pyruvic acid, and so on and so forth, which is what happens in the fasted state, by the way, right? When you break down glycogen, you can convert it back to this molecule and then take it through glycolysis to make energy, right? But in the fed state, you're basically going to just form glycogen because you have enough glucose in the body already, right? Your cells are happy. They made the energy they need, store the rest of it in glycogen now. In your fasted state, this is what's going to get broken down, right? And what happens is the liver especially has the enzymes to physically take glycogen all the way back to glucose, and then that can be put back into the blood and shared with the rest of the body. This is how the brain can still have access to glucose, right? So that's happening now in your fasted state. When you start to break down your nutrient stores, you could still provide the body with glucose because the liver can break down this glycogen and share it with the body. Other tissues cannot do this. Skeletal muscle, for example, can break down glycogen, but it cannot form glucose. It literally lacks the enzymes to do this. So instead, it will break down glycogen to this glucose 6-phosphate, and that could then be just used to make energy then, okay, not to make glucose. It just makes energy for itself. And that's why Dr. Woods referred to the skeletal muscle as selfish tissue, right? It's making energy for itself. Compared to the liver, it's the more kind of friendly sharing organ. It's going to share this glucose with the rest of the body. Okay, any questions on that? Okay, so that's great. We've made glucose, shared it with most of the tissues. They made their energy. The rest of it got made into glycogen in the liver and the skeletal muscle. Then what? What's the last thing that happens if you still have glucose remaining? Fats, right? Exactly, right? So again, you never want to waste nutrients. So the rest of it gets converted, especially into glycerol that can combine with fatty acids, and that can then be used to form your triglycerides, right? These are your main fat molecules right over here, triglycerides, okay? And these will be kind of put into your adipose tissue, right? That's your fat tissue, okay? And unlike glycogen, remember I said glycogen has a very limited capacity to be stored, right? There's so much space in the liver and skeletal muscle, but eventually it will get filled up with that glycogen. This is why triglycerides are helpful because there's virtually an unlimited capacity to store fat, which is why people can get up to 400 or 500 pounds and still be living somehow, right? That's the idea. Their adipose tissue can just continue to expand and expand and expand until you're morbidly obese, and it's just not fun. Okay, so those triglycerides get stored in the adipose tissue. Okay, so that was all carbohydrates. Any questions on that? Okay, let's talk about proteins. And thankfully, proteins and lipids are a little bit less complex than carbs for the most part. Okay, so again, what are proteins? Where do they come from? Well, they come from animals and plants, right? Animals and plants have the proteins that we do. They have enzymes and transporters and receptors and cytoskeleton components, all made up of proteins. So when we eat those meats or those plant sources and we digest those proteins, we break those amino acids apart, and that is what we can then use to make our own proteins for our own body. Okay, so you might have remembered from high school bio, we have 20 different amino acids. We have what are called essential and non-essential amino acids, right? So again, remember, essential are the ones you have to get from your diet, right? Your body cannot make them, which is why we call them essential amino acids. You have to ingest them some other way. Non-essential meaning your body can make them. It's non-essential that you ingest them through your diet. Okay, and for the most part, we call like large kind of chained peptides or proteins polypeptides, right? So thinking back to what we talked about with the carbs, right? Those are basically sugar units attached to each other. Same idea with proteins. These are basically amino acids that are attached to each other, right? And just like you had disaccharides and trisaccharides and monosaccharides, you have dipeptides and tripeptides. And polysaccharides, you have polypeptides, right? These would be like the equivalent of complex carbs, basically, right? So lots and lots of amino acids attached to each other. What can we absorb when it comes to amino acids? When it came to carbs, it was monosaccharides specifically. What can we do with amino acids? Yep. Absolutely. We can absorb single amino acids. That's nice and easy to remember, just like monosaccharides. Anything else? Absolutely. So it's not ideal. Our body would prefer to have single amino acids, but our body can technically absorb di and tripeptides as well. The point is you cannot absorb longer polypeptides. Okay, so anything more than 3 amino acids are not absorbable. That means you have to break these down into smaller and smaller units so that you can generate the single or double or tri amino acids, basically. Okay, so same idea. Let's start off with what's happening in the mouth, right? So you've just ingested a nice steak. Oh, steak. Love steak. Or you have chickpeas. Anything happening in the oral cavity. You have any protein digestion in the oral cavity. I'm really hungry, by the way, in case anyone doesn't realize. No, a lot of people are shaking their heads, right? So we do not have any digestive enzymes in the oral cavity, right? So no pepsin or anything else that's being secreted in the oral cavity. So basically, when it comes to the mouth, it's just salivary amylase and another enzyme, lingual lipase, which we'll talk about with the lipids. Okay, but nothing involved in protein digestion here. Okay, so no chemical digestion, but again, keep in mind, you do have mechanical digestion, right? The chewing action of your mouth to physically break down food into smaller components. Okay, stomach, you have the enzyme pepsin. Where exactly is pepsin coming from? Does anyone remember the specific cell type? Okay, again, it's multiple. Oh, go ahead. Yes, absolutely. Chief cells, right? So again, remember your stomach has, well, I'll just show you. It's mucous neck cells, chief cells, and parietal cells. These are your exocrine cells, right? They're the ones secreting things into the lumen of the stomach, right? So chief cells are secreting that pepsin to start with protein digestion. You'll note here it says pepsinogen, even though we said it was pepsin is the enzyme. What is pepsinogen specifically? Yep. Absolutely, yeah. It's the inactive form of pepsin, right? We refer to it as a zymogen, or the inactive form of an enzyme. Why would we secrete an inactive enzyme? Why would we not just secrete pepsin? What's the big deal here? Why does it have to get activated? Yep.
Start to digest itself kind of thing, like that organ.
Yeah, exactly. What does pepsin do? It digests proteins, right? It's digesting polypeptides into smaller and smaller peptides. Do we not have proteins in our cells? Yeah, we do, right? We have the receptors and ion channels and transporters, everything we just talked about previously. Those are all proteins. So if pepsin was secreted, activated, it could literally go back to the stomach cells and digest those proteins. You would literally digest the cells that made this enzyme in the first place. You would digest your own body. That's a big no-no. So what happens is it gets secreted as pepsinogen into the lumen of the small intestine, and then something specifically is going to activate it into pepsin. Does anyone remember what that specific something was? Absolutely, right? So the stomach acid, right? So remember all these different functions of the stomach acid. Here's the second function right over here. It's going to activate, it's going to literally cut pepsinogen into its active form pepsin. And now that it's in the stomach and there's food around, it can digest the food in our diet and not the cells of our body, right? So this can now begin protein digestion. And keep in mind, the stomach acid is also going to help with protein digestion as well, right? It unfolds proteins. Right, you remember from high school bio, you learned about tertiary and quaternary structure of proteins, right? That folded structure. If it's folded up, pepsin's going to have a pretty hard time cutting up the protein bonds if there's like some amino acids kind of like deep within that fold. So the stomach acid will literally unfold the protein, and now pepsin can cut up that unfolded protein, and that makes it a lot easier to start chemical digestion. Okay, so that's happening in the stomach. Pepsin is going to break up those larger polypeptides into smaller peptides. Next up is your small intestine. So let's start off with what's happening in the small intestine itself. Do we have any enzymes being made in the small intestine that are involved in protein digestion? The answer is yes. Again, it's the brush border enzymes again. Right, so you have two specific protein digesting enzymes, aminopeptidase and dipeptidase. And again, just like lactase and sucrase and maltase, they're named for exactly what they do. Aminopeptidase is going to break off an amino acid from the amino terminus. We're going to see what that looks like in just a second. Dipeptidase, what do you think this does? It's going to break up dipeptides. It's right in the name. Keep in mind, the small intestine also gets help from the pancreas. Okay, so the pancreas secretes a bunch of enzymes: trypsin, chymotrypsin, and carboxypeptidase. Unfortunately, trypsin and chymotrypsin have crappy names and they don't really make any sense to what they digest. So you just have to memorize that. They're protein digesting enzymes. But carboxypeptidase is named for exactly what it does. It's like aminopeptidase. It's going to cut one amino acid off at a time from the carboxy terminus of a polypeptide. Okay, so again, we're going to see what that looks like in a second here. Okay, so keep in mind, starting off with these aminopeptidase and dipeptidase enzymes, these are found in the small intestine. Remember the brush border? These are brush border enzymes, just like lactase, sucrase, and maltase. These are physically found within the membrane of the small intestinal cells. Okay, they are part of that brush border again. Aminopeptidase, dipeptidase. You also have a third one, enteropeptidase. This is important for activating the pancreatic enzymes, which we'll see here in just a second. Okay, so here are your pancreatic enzymes, trypsin, chymotrypsin, and carboxypeptidase. Oh, but hang on a second, your pancreas is actually not secreting trypsin, chymotrypsin, and carboxypeptidase. They're secreting trypsinogen, chymotrypsinogen, and procarboxypeptidase. What are these three exactly? Are they enzymes? Yep. They're proteins, right? So again, they're inactive enzymes. There are zymogens. Okay, all enzymes are proteins, but these are specifically inactive enzymes. Why do we do this? Same idea as pepsinogen. We have proteins in our pancreatic cells, right? You do not want these enzymes to digest the pancreatic cells themselves. So instead, same idea. You secrete them as the inactive form. They get put into the small intestine, and then they can start to digest the proteins of your diet and not the proteins of your actual cells, right? You don't want to digest your actual cells of your body there. And the last thing I want to say about the slide is, again, these are coming from your acinar cells specifically. Okay, so not ductal cells and not the endocrine cells, right? Those alpha cells or beta cells that make insulin or glucagon. These exocrine secretions are coming from the acinar cells specifically. Now, let's talk about that enteropeptidase, that last brush border enzyme, right? So in the stomach, what activated pepsinogen? It was the stomach acid, right? Now, in the small intestine, you do not have stomach acid, right? So you have to have something else that's going to activate your inactive zymogens, right? So that enteropeptidase, also called enterokinase, which is another brush border enzyme, is going to cut trypsinogen specifically into the active form trypsin. What does trypsin do? It's a protein digesting enzyme, right? It's going to start to digest proteins. And look at that. There's three other proteins that were kind of secreted along with it from the pancreas. Chymotrypsinogen, procarboxypeptidase, and prophospholipase, which is involved in phospholipid digestion. These are proteins. Trypsin will literally cut them. They will digest them, and it'll end up activating them. You end up creating the active form of the enzyme, chymotrypsin, carboxypeptidase, and phospholipase. Okay, so specifically, trypsin, chymotrypsin, and carboxypeptidase, they're involved in protein digestion. And so they're going to be the ones that are digesting the proteins in your diet. Okay, so here's a summary slide of all the protein digesting enzymes, right? So pepsin, again, is coming from the earlobe. That's the earlobe, right? What'd you say? Not the pancreas, something else. Pepsin? Go ahead. Yeah, stomach, right? So remember, gastric pepsin, it's coming like pepsinogen coming from those chief cells as we talked about earlier, right? And then also you have help from the pancreas, trypsin, chymotrypsin, and carboxypeptidase. And you also have from the small intestine, aminopeptidase, which is a brush border enzyme, and dipeptidase, which is not shown here, also a brush border enzyme. Okay, it's going to cut up dipeptides again. Okay, so keep in mind, you can actually subdivide these further into classes of protein digesting enzymes, right? So they're all peptidases, right? Because they're all involved in digesting peptides, just like lipases are digesting lipids. But where they cut can further kind of separate them out, right? So endopeptidases cut in the middle of polypeptides. Exopeptidases cut at the ends, the exits of the polypeptides, if you want to think of it that way. Right, so here's that aminopeptidase and carboxypeptidase I talked about before, right? Aminopeptidase is cutting off one amino acid from the amino terminus specifically. Carboxypeptidase is cutting off one amino acid from the carboxy terminus. Okay, so these are named really, really nicely. right? Amino for the amino end, carboxy for the carboxy end. Now here's the thing. This is handy, right? Because right now we're generating single amino acids. Think about what we talked about at the very beginning. These are the most ideal things to absorb, right? So this is awesome, but this would take forever if we're cutting off one amino acid at a time and we have one very long polypeptide. So these endopeptidases can cut in the middle, and then look at that. You generate some more carboxy termini and more amino termini. And now these enzymes have more ends to work with, right? More things to pull off, and that speeds up the process a lot. Does that kind of make sense? Sort of? Okay, sweet. All right, so you've now cut up your polypeptides. Ideally, you've generated single amino acids or maybe a couple of di and tripeptides. Then what? It's in your small intestine. You want to now absorb them into the body, right? So same idea with the carbohydrates. You have your intestinal cells. On the luminal membrane, you have, look at that, an amino acid sodium symporter, right? It's a similar idea to the glucose or galactose symporter. By the way, you've seen this transporter as well before, right? Remember the kidney and the proximal tubule? This is the one that's going to be reabsorbing your amino acids from the proximal tubule lumen. Right, so same idea, regardless of what the concentration gradient for amino acids is, sodium has such a high concentration gradient that it's going to physically drag all the amino acids with it, right? This helps to maximize amino acid absorption into the body. Now you have amino acids in the cells of the small intestine. And again, keep in mind, you can also absorb di and tripeptides as well with this symporter here. I think Dr. Wood said, don't even worry about the hydrogen part. Just keep in mind, you can absorb, bless you, you can absorb di and tripeptides. Once those di and tripeptides are in the cell, again, it's not really handy to have 2 or 3 strands of amino acids because your body wants to decide how to put those amino acids together, right? So what it's going to do once they're in the cells is you're going to further break down these di and tripeptides into single amino acids if necessary, right? So you have some more peptidases available there to break off the remaining di and tripeptides. And again, it's no use if these amino acids are just sitting in these cells, they have to get into the blood. So again, on the basolateral membrane, you have an amino acid uniporter, and that's about the extent that you have to know about that, right? Again, keep in mind, you probably have several transporters for each amino acid, but you don't have to know that, right? And now it can get put into the blood and distributed throughout the whole body. And then again, keep in mind, you also have that sodium potassium ATPase, right? That's keeping sodium nice and low in the cells for these transporters to keep working. Okay, so again, summary slide now, looking at amino acids, right? So now the amino acids are in the blood. They were brought to the liver by that hepatic portal vein. The liver said, okay, everything's good. Let's shuttle these amino acids onward to the heart. The heart pumps these amino acids, and now they're perfectly fine in the circulation. What are amino acids used for in the body? Yep. Yeah, exactly, right? It's like we digested proteins from our diet to make amino acids for our body to then make the proteins that we need, right? You don't know what proteins are in plants or animals. We may not want those proteins. So instead, we break them up, we make the building blocks, and now we have access to all those building blocks to make whatever proteins we want to make for our body. We need to make more myosin and actin for our muscle, we can do that. Right, so amino acids on this slide here, it says it's going specifically to the skeletal muscle, but keep in mind, it's going to all cells of the body, right? All the cells of your body have proteins, but the skeletal muscle especially has a ton of protein, right? All that actin and myosin and the M-line and Z-line and titin and all that, those are all very important proteins for muscle contraction. This is why if you want to build muscle, they say drink protein shakes while you're doing a lot of exercises, right? Because then you maximize how many amino acids you have in your circulation to build up those proteins then. Okay, then what? You have all the proteins you have or need in your body, then what? Do you just get rid of the extra amino acids, or what do we do with them? Go ahead. Exactly, you're gonna store it as fat, right? Again, you do not want to waste nutrients, right? So those amino acids can be converted to fatty acids. That can combine with glycerol, which you can get from glucose being converted to glycerol. Excuse me, and then those two could be put together to make triglycerides, right? So again, long-term fat storage. Do not waste nutrients because you don't know when the next meal is going to be coming around. Obviously, that's not an issue for us because we have access to food pretty readily. But again, these systems have evolved from caveman days when food was not easily accessed. Does that make sense? Any questions on proteins? All right, let's finish off with fats. Okay, so again, We talked about how proteins are coming from animal and plant sources. They're large proteins like polypeptides. Carbs were complex carbs or disaccharides. When it comes to lipids, nice and easy. Basically, all you have to know is the main lipid molecule, which are triglycerides. Okay, so here is a triglyceride. It has a glycerol backbone, which is these kind of like 3 carbons over here, and 3 fatty acids. Okay, so that's why you get the name triglyceride. Tri for the 3 fatty acids. Okay, ultimately, we want to break these down, and again, small units that can be absorbed into the body. Okay, any lipid digestion taking place in the mouth? No. Any enzymes being secreted in the mouth that are involved in lipid digestion? Yes, right? So you have lingual lipase, but it is not activated in the mouth. Okay, so our salivary glands will secrete this. And again, remind me, which salivary glands specifically are going to be involved in this? I see you mouthing it. Yeah, you have the first one. Yeah, exactly. The parotid gland and yeah, submandibular gland, right? So again, make these connections, right? This is a study tip for you guys. As you're studying lecture 6 or lecture 5 of digestive phys, think back to what you talked about in lecture 1, right? Where those enzymes are coming from in the first place, right? So the parotid gland, actually I have a slide over here. The parotid gland, right? It's very heavily producing watery saliva with tons and tons of enzymes. The submandibular gland also making enzymes, but to a lesser degree. Sublingual gland not doing anything, right? When you're eating, this is not secreting any enzymes. It plays no role in enzyme or chemical digestion. Okay, so here's the thing. Lingual lipase was made in the mouth. It was not active in the mouth, but once it's swallowed, it's brought to the stomach, and that is where it gets activated. What's so special about the stomach that activates lingual lipase specifically? Absolutely, right? It's the same idea. It's like pepsin. This enzyme works at a very specific pH. And if it's working at an acidic pH, right, in the stomach, you're looking at a pH of around 2, it's not going to work in your mouth because your mouth is not that acidic, right? So the stomach acid will activate the lingual lipase because of that low pH. This is an enzyme that works well in a low pH environment. Keep in mind, you have another lipase in the stomach as well, gastric lipase. Okay, so 2 lipases are activated in the stomach that are involved in lipid digestion. Where did this gastric lipase come from? Mucous neck cells, chief cells, parietal cells, G cells. Not G cells. That was a bit of a trick answer there. Yeah, it's chief cells, right? So chief cells are making both pepsinogen and gastric lipase. So there's a study tip for you, right? The enzymes are coming from your chief cells, right? G cells are making an endocrine secretion. That's the hormone gastrin. Okay, so keep that in mind. That's an easy trick that Dr. Woods could put on a question there for you, right? You might have an option that says, what are exocrine secretions coming from the stomach? She might put, a secretion coming from the G cells. And it's like, no, that's an endocrine cell. Okay, it's making the hormone gastrin. Okay, so you have lingual lipase from the oral cavity, specifically from the parotid gland and the submandibular gland. And you have gastric lipase coming from the chief cells from the stomach. Okay, so 2 enzymes in the stomach that are involved in lipid digestion. Okay, so these are going to start to break up triglycerides. What exactly do they do? Well, this is where you have to kind of think back now, right? So just like how the complex carbs were broken down by amylase in a specific way, or the disaccharides are broken down by the brush-border enzymes in a specific way, lingual lipase and gastric lipase do a very specific thing with lipid digestion. Right, so keep in mind, we're starting off with triglycerides, a glycerol and 3 fatty acids. What happens is these lipases will physically pluck off, they'll literally pull off the free fatty acids, right? So you're going to go from a triglyceride ultimately to a monoglyceride and those free fatty acids. Okay, these are what are absorbable. Okay, just like how monosaccharides were absorbable, just like how single amino acids and di and tripeptides were absorbable, triglycerides are not absorbable. Monoglycerides are absorbable. Free fatty acids are absorbable. And again, it has to do with their size primarily, right? Triglycerides are way too big to actually move into cell membranes. Okay, so quick question. We have 2 enzymes here, right? Surely that means stomach digestion of lipids is efficient. Is that true? No, right? Again, keep in mind, lipids are hydrophobic, right? They do not like water. And guess what? Our stomach is a very watery environment. So these lipases are going to have a difficult time breaking these lipids apart because they're going to end up forming large lipid droplets, right? They're forming a shape that basically minimizes the interactions of that droplet with the surrounding watery environment. So these lipases can really only act at the surface of a large droplet. That's not a lot of surface area if it's one large droplet, right? So just keep this in mind from now. These lipases are not very efficient at digesting these lipids in the stomach. Okay, then you're in the small intestine. What enzyme is going to be involved in lipid digestion specifically in the small intestine? Yep. Absolutely. So you have pancreatic lipase, right? So just like lingual lipase, gastric lipase, now you have pancreatic lipase. They're all named for exactly what they do. This is coming from the pancreas. Again, what specific cell type of the pancreas? The acinar cells, right? It's the acinar cells, those clusters of cells that are making that exocrine secretion. They also make another thing called colipase, right? So again, this is not an enzyme. You've heard me talk about this in tutorials. This is kind of a protein helper. It's going to help with lipid digestion. It's going to help lipase do its job, but it does not digest lipids itself. Okay, so it's not an enzyme. Okay, and then last thing, this is probably the most important part and why the small intestine is so efficient at lipid digestion. You have another secretion, not an enzyme. It's a secretion, an exocrine secretion, bile. Okay, the bile solution coming from the liver. It contains something called bile salts that's going to help to coat the lipid droplets and keep them small. We're going to see what that looks like in a second here. Okay, so again, pancreatic lipase coming from your acinar cells. You also have colipase coming from the acinar cells as well. And it originally starts as, again, an inactive protein. But again, keep in mind, once it's activated, it's still not considered an enzyme. Okay, it's a protein helper. And then lastly, you have another thing that we're not going to spend too much time talking about, but it's another enzyme called phospholipase. Okay, so keep in mind, if we're ingesting food, whether it's plants or animals, they are made up of cells. And every single one of our cells, whether we're a plant or an animal, has a cell membrane on it. It has phospholipids. So we have an enzyme, phospholipase, that's going to break down those phospholipids. And guess what? Now we can absorb those and help make our phospholipids for our cell membranes. Okay, so keep in mind, I'm not going to go through all of this here, but bile is coming from your liver, right? So specifically, it's coming from the hepatocytes, these little pink cells all around the whole liver, right? They're going to secrete that bile into specific ducts called bile canaliculi. Those come together and eventually will join the secretions from the gallbladder, another tissue where bile is stored. Altogether, all that bile is going to come down into the small intestine and help with lipid digestion. I glossed over a lot of details there, but I don't want to spend too much time on those details. Those things you could just read over, right? It's like the names of the ducts, for example. Okay, so keep in mind, the gallbladder also plays a role here, right? So the gallbladder does not make bile, but it stores bile. Okay, so the liver will make bile, they'll secrete it into its ducts, and eventually those bile solutions will end up going back to the gallbladder and kind of just stored there for later on when you're actually eating. And in the process of being stored there, water will actually be actively removed from that bile solution. That makes it much more concentrated. That means there's more bile salts within that solution, right, relatively speaking, which is going to help with lipid digestion in the small intestine. Once you've eaten food, bile is going to be leaving the liver. The gallbladder is going to contract and release its bile, and ultimately they're going to enter into the small intestine. Right? They join up with the pancreatic duct and eventually will just secrete things into the first part of the small intestine, that duodenum. Keep in mind you have a sphincter here as well to make sure that things are moving in one direction and not backwards. Right? You don't want acid or enzymes accidentally going up these ducts and digesting parts of your cells again. Right? So you have a sphincter there to make sure it's all going in one direction. And what exactly leads to bile getting secreted? Well, you have a hormone called CCK, right? So I'm not going to go into those details. Again, that's just a straight memorization thing, but you have cells in your small intestine called CCK cells. They release this hormone into the blood, and it's going to act on the liver and the gallbladder and the pancreas to cause the release of those exocrine secretions. right? So everything we talked about, all those like trypsin, chymotrypsin, or pancreatic amylase, bile solution, bile from the gallbladder, that's all being triggered by the hormone CCK from the small intestine, right? So you can now tie in what you learned about the whole intestinal phase of gastric regulation, right? Once food is in the small intestine, you signal to all of these organs to release those substances. And that is what then helps to digest those nutrients. Does that all make sense so far? Okay, talk about that. So what exactly do bile salts do? How is lipid digestion handled? Well, you're gonna have, again, lipid droplets that have made it into the small intestine. You have your segmentation patterns in your small intestine, right? That's gonna really mix up the lipid droplet and break it apart. Now, normally, again, lipids do not like water, and a lot of the secretions in your stomach and your small intestine are very watery, right? So normally these lipid droplets, when they're small, they'll want to join back up together and form larger lipid droplets so that they can interact with each other and get away from the watery environment. But the bile salts from the liver and the gallbladder prevent this from happening, right? So you have bile salts that are literally going to coat the lipid droplets and keep them nice and small. What does that do? It means there's way more surface area available now for pancreatic lipase to break down those triglycerides, right? So that's the difference. It's not that pancreatic lipase was better than gastric lipase or lingual lipase. It's the fact that these droplets are way smaller and more numerous that increases your surface area. That means you have way more potential for these enzymes to break down those triglycerides. Same idea, you're going to break them down to your monoglycerides and free fatty acids. These are absorbable. Okay, and one other thing is in the small intestine, you have that protein helper, colipase. All it does is it physically pushes the bile salts apart and allows that lipase to access the lipid droplet to digest the triglycerides. That's the extent that you have to know about that. Okay, so now you have a bunch of monoglycerides and free fatty acids. Then what? Well, again, you want to absorb them into the body, right? So first thing that happens is it moves into the intestinal cells, right? So the cells of the small intestine, those absorptive cells, they are kind of put back together into triglycerides because those are the main fat molecules. But here's the thing. They are fats. Are they going to be happy being put into the blood? Blood is watery, right? Think back to the endocrine section, all those hydrophobic hormones that we talked about. They had to be put into, or they had to be bound to plasma proteins to circulate in the blood, right? Same idea here. You can't put these fats in the blood as is. You have to put them in some sort of a protein shell. That's what these chylomicrons are, right? So they're basically proteins that have a hydrophobic component that can face the lipids and keep them nice and happy, and a hydrophilic component that can face the watery environment of the blood vessels. Now they're soluble in the blood. They can be put in the blood and they're perfectly happy being in that blood then. They could circulate in the body. One other problem, though, is that these chylomicrons are huge. And they can't actually physically fit in between the pores of your blood vessel cells, those endothelial cells, right? So they have to first be put in lymphatic vessels. These vessels have much larger gaps in between them, and now they can be pumped across the body. Eventually, these lymphatic vessels will drain back into your normal blood circulation. And at that point, they can be distributed to all cells of the body. Does that make sense? Okay, lastly, summary slide again. So now you have a bunch of these fats in your circulation. What are fats going to be used for in the body? Keep it nice and simple, right? They could just be used to store fats in your body, right? Literally taking the fats from your diet and storing them in your body as fats. If necessary, though, you could use them for energy. For example, that's what ketogenic diets kind of take advantage of, right? So if you're not ingesting enough carbohydrates, you don't have enough glucose, you can use fatty acids and glycerol to make energy, right? But under normal circumstances, if you're eating a balanced meal, chances are you're going to have enough glucose to supply your cells, in which case these fats are just going to be used to store fats for your body for long-term storage. Okay, keep it nice and simple. Does that make sense? Okay, that is basically everything I have for digestive physiology. Obviously, I didn't cover everything there. I left like the fasted state and some of the other components like the cephalic phase, gastric phase, and all that. But this, I think, is a good overview of basically everything that we talked about from start to finish. So before we move on to reproductive, I'll open the floor to any questions if there are any. Yep.
Can you just quickly go over how, like, what enzyme like kind of separates the bile salts in that? I think.
Yep, not an enzyme. It's a colipase. Okay, so colipase, remember that was one of the secretions coming from your pancreas, your acinar cells, not an enzyme. Okay, so the distinction there is that enzymes are going to be involved in digesting lipids, or rather, like the lipid digesting enzymes are digesting lipids. That's how we define them. Colipase is not an enzyme, which means it does not actually digest lipids. Okay, so what it does is obviously pancreatic lipase, I told you, has to actually get to the triglycerides to break them up, right? They have to physically rip off those fatty acids from the triglycerides. They can't do that if the bile salts are in the way. Right, so what colipase is going to do is it's basically going to come in and do this. It's going to push the bile salts apart, and now your lipases have access to those triglycerides that can break them apart. Does that make sense? Yeah, you're welcome. Any other questions? Okay, so we're about an hour and 10 minutes in. Are we good to move on to reproductive physiology? Okay, before we do that, let's take a quick 2-minute break. I need some water, and we will get back here in by 11:13. So I was severely dehydrated. What hormone was getting released from my posterior pituitary? ADH, yeah. What is that ADH going to be doing? It reabsorbs water, right? From which segment of the tubule? You're collecting duct, absolutely. That's another study tip for you guys. Make things relevant for you in your own life, right? So I know like this is so nerdy, but every single day I wake up, the first thing I think of is, my blood pressure is probably pretty low right now because I don't have that much water in me and all that. Or my sympathetic nervous system is activated when I'm running for the bus or something stupid like that, right? So I'm thinking of like everything we talk about, right? Like how my heart rate is working, how the action potentials are increasing in my SA nodal cells. Like make those connections. I know it sounds kind of silly and nerdy, but it really helps to remember things as you're studying for these exams. And we're lucky because physiology is literally as applicable as it gets to our own lives, right? So making those connections to your own body is so, so helpful. And pretty much everything we talked about, with the exception of reproductive physiology, because obviously, males are not, they don't have ovaries and females don't have, testes, and we're not getting pregnant all the time. But everything else that we're talking about in this class applies to pretty much every single thing that happens to us in our day-to-day lives. Even motor and sensory physiology. I know that's a bit of a tricky unit for everyone, but make those connections. I'll give you a quick example. So we talked about photoreceptors with Dr. Everling. He told you that the fovea, which is like the center of your field of view, has the most cone photoreceptors. Does anyone remember what cone receptors do specifically? Anyone started studying that yet? Yeah, it's your color vision, right? So I'll give you a cool example here. I don't know if you can really tell on the slide here, but what color? Actually, this is a terrible example. Let's do this. Actually, that's way too bright too. Okay, this is perfect. Okay, so if you look at this head on, you can see that lipid droplet is yellow. If you look at it from the side though, if you were to stare at the wall, I can't see what that color is at all. In fact, I could barely tell what any color is with the exception of this kind of pinkish color right over here. Right? That's because your cone receptors are mostly found in your fovea. You have very few cones in the periphery of your vision, which is why you can barely see color. What do you have in your periphery? Well, you have a lot of rods, right? So this is just a bright image to me. In fact, it's a lot brighter here compared to me just looking at it dead on, because the photoreceptors there are more sensitive to light than they are color, right? So make those connections in your own life, because now you're never going to forget that if you try to make it relevant to yourself. Anyways, that's a bit of a side thing here. All right, let's get back to reproductive physiology. Again, I'll start off with Q&A. Is there anything specific that anyone would like me to cover to start off with? Right, so here was my kind of to-do list for reproductive physiology: spermatogenesis, oogenesis, folliculogenesis, and ovulation, all the effects of LH and FSH, the uterine events, how estrogen and progesterone play a role in that, and fertilization and development. Right, so is there anything anyone would like me to start with to kick us off? Or do we just want to go with that order? All right, spermatogenesis. Are we all good with that? Let's do spermatogenesis. Okay, so again, if you haven't had a chance, please do that last tutorial activity that I had planned for our final tutorial. Right? I know we did that Kahoot, but the activity is actually really, really useful. It has like a little puzzle piece thing where you can basically arrange the order of spermatogenesis and oogenesis. And it really, really is helpful for studying that whole process. Okay, so let's start off with spermatogenesis. I'm bringing that up, by the way, because I'm using those exact same slides here. I'm also going to spend a bit of time here talking about mitosis versus meiosis, just in case you see a question on that on the exam. I don't know how much Dr. Bell wants you to focus on that, because she spent like 2 minutes talking about mitosis versus meiosis, but I'll spend a little bit of time here as well. Okay, so starting with spermatogenesis, right? So keep in mind, we're talking about a biological male here, right? So before the baby is born, they have their structures, right? Their testes and their penis and everything that they need, but nothing is happening before birth. Nothing is happening after birth either. Things only start at puberty. So what exactly is there? What is present before puberty? Well, you have these stem cells, basically, the spermatogonia, that are present, but aren't really doing anything. They're dormant, basically. Right? So at this point, what do they look like? Well, they basically have 23 pairs of chromosomes. Okay, so I have on the slide here 4 pairs depicted. I couldn't draw 23 here, but you can imagine there would be 23 pairs of chromosomes. Why am I saying pairs? Where does this pair idea come from? Like, why isn't it just 46 chromosomes? Why do we call it 23 pairs of chromosomes? No idea? What do we need to be born? Do we need just our dads or just our moms? No, right? So the 23 pairs is coming from the fact that one set of those 23 chromosomes is coming from your paternal, like your father, biological father, I should say. And one set of those 23 chromosomes is coming from your biological mother. Okay, so you have 23 sets of chromosomes. You have chromosome 1, chromosome 2, chromosome 3, chromosome 10, chromosome 11, so on and so forth. And you have 2 of each of those. Okay, 1 from dad and 1 from mom. That's how you get those 23 pairs of chromosomes. Okay, so these cells, these sperm cells, these stem cells as they are right now, are just like any other cell in the body. They have 23 pairs of chromosomes, all the genetic material that makes up us. Okay, but again, keep in mind, these are going to be a little bit more different because they undergo meiosis compared to any other cell type in our body, which is all happening through mitosis. Okay, so we're going to talk about this here in just a second. All right, I forgot. Actually, I annotated these. Okay, so 23 pairs of chromosomes. Here on out, I'm only going to show one pair because this was a real pain. I had to like manually draw these out. Okay, so baby boy is born, nothing happens. Then he hits puberty around 9, 10, 11, 12, 13, whatever age it is, doesn't matter. Okay, then what happens? Well, those dormant spermatogonia, right, the stem cells, which have kind of just been hanging out for all this time, they start to undergo mitosis. Okay, so what is mitosis? It is the generation of genetically identical daughter cells. Okay, so you're starting off with one spermatogonia, and keep in mind, males are going to have several of these. They're not going to just have one. They're going to have probably millions or way more than that even. What exactly happens in mitosis? Well, every single one of those 46 chromosomes are going to get duplicated. Okay, now here's the best part of biology and why things get a little bit confusing. And again, I don't think Dr. Bell needs you to understand this as much, but it's helpful for you to understand when you're undergoing meiosis. Even though we've duplicated every single one of our 46 chromosomes, we still call it 46 chromosomes. It has to do with the way that we count chromosomes. It's based on this kind of middle section over here called, I think it's called the centromere or centrosome? Centromere, I believe. Right? The fact that you have these two arms attached at the same centromere means that there's only one centromere. And we count that as the chromosome. That's how we count chromosomes. Okay, that's an aside. So how exactly do we count these then? This is different from this, obviously. It's duplicated. We say that there are 2 chromatids. Okay, so right now we've gone from 46 chromatids to 92 chromatids. Those chromatids are what get pulled apart in mitosis, right? So the duplicated DNA, you're literally pulling apart the duplicates and separating them out into 2 new daughter cells. And guess what? You're now exactly where you started off at the beginning, right? You have the exact same genetic material, but now duplicated into genetically identical daughter cells. Okay, this is what mitosis does. That's the gist of it. And of course, you might remember from high school bio, you had all of like, you know, prophase, metaphase, anaphase, telophase, all that crap that you don't need to know for this course. But that is mitosis. That's just for the purposes of making more spermatogonia, right? These are not sperm cells. These are sperm stem cells. And these undergo mitosis for the purposes of constantly having access to these cells that can make sperm cells. But you do not want all of them to make sperm cells because once they undergo meiosis, they can't do mitosis anymore. They can't replicate and make more of themselves. So these cells are constantly going to be making more of themselves, but a subset of them will undergo meiosis, and they're the ones that are going to develop into the sperm cells. Okay, so this is very different now. We're talking about meiosis. So those spermatogonia, which begin meiosis 1, we now classify as primary spermatocytes. Okay, so these are no longer capable of doing mitosis anymore. They have begun meiosis 1. And notice I'm saying begun, they haven't completed meiosis 1. They've just started it. Same idea in mitosis. You're going to duplicate all of your DNA. Okay, so every single one of those chromosomes, all 46, are going to get duplicated. But by the same logic, we still refer to it as 46 unique chromosomes because they all are attached by that one centromere, I think it's called. But we refer to them as each having 2 chromatids. Okay, so that's what we call these 2 arms, basically, right? So in total, you're going to have 92 of these, right? Because you have 46 chromosomes to start, and if you duplicate all of them, they're each going to have 2 chromatids each. Okay, so 46 times 2 is 92. 92 total chromatids. At this point, we still call it diploid, right? Because you still have dad's chromosome and mom's chromosome in that sperm cell there, right? So there's 2 sets of every single one of your chromosomes. Chromosome 1, 2, 3, 4, 5, all of them. You still have the 2 pairs there. and they're all duplicated as well, which is what gives you the chromatids. Now, this is what's cool about meiosis. Unlike mitosis, which gave rise to genetically identical cells, meiosis is all about creating genetically unique cells. Because guess what? I don't want my offspring to be identical to me. Holy crap, that would suck. I'm a terrible human being. But if there was a virus that attacked my genetic material, that means my offspring would also get screwed, right? So this idea of creating genetically unique cells in meiosis helps to ensure or kind of prevent that from happening. It makes it more likely that he can survive that virus if that virus was attacking a specific part of my genetic material, for example, right? So we want to make genetically unique offspring so that if there's some major selection force, right, some major disease or some major disaster that only affected a subset of people with certain genes, they will die off, but not the whole human race would, right? You're still going to have the human race continuing on. They're the ones that have the genes that will continue on and be fine with that disease in place. Does that kind of make sense? Okay. Right, so how exactly does that happen? Well, this is that idea of crossover, right? So again, you might have learned this in high school bio. You don't need to know the details about it. But the point is, basically the genetic material from dad's chromosomes are going to swap with some of the genetic material from mom's chromosomes. And what you end up with are, let's say this is chromosome 1. All of a sudden, chromosome 1 now has kind of a mix of dad's and mom's genes. It's not completely identical to dad and mom anymore, right? So now it's genetically unique material. The offspring is not going to be completely identical to either mom or dad anymore. Okay, so then that primary spermatocyte will finish meiosis 1, and that's then again give rise to 2 secondary spermatocytes. Okay, so here we've divided. This is a big distinction between meiosis and mitosis here, by the way. What we've done is we've basically divided dad's chromosomes and mom's chromosomes from each other, right? They're the homologous chromosomes, if that rings a bell to anyone, right? So at this point, we now have 23 chromosomes in one cell here and 23 chromosomes in one cell here. One that has the dad's genes and one that has the mom's genes. But again, keep in mind, they're not all dads and all moms now because they had crossover taking place. It's kind of a mix and match event taking place here. At this point, though, they are 23 chromosomes. We refer to this as haploid, or just N at this point, right? Diploid was 2N, haploid is N. Question for you, though. Do you want this to merge with the female ovum? This is still duplicated genetic material. Even though it's 23 chromosomes, you still have 2 chromatids for every single chromosome, right? It's still duplicated. So you have to separate these out as well so that you have single strands of chromosomes. That is where meiosis 2 comes in, right? So meiosis 2 will again now split off the chromatids from each other, and now you have 23 single strands of unique chromosomes for each of these new cells that we now call spermatids. Okay, so this happens now times 2, right? You have 2 cells that come from the secondary spermatocyte, and remember, you had 2 secondary spermatocytes for every primary spermatocyte. Okay, so in total, how many gametes do we have for every round of meiosis? You have 4. Okay, 4 gametes, 4 spermatids for every single primary spermatocyte. Does that make sense? Okay, this is a very big distinction between males and females. Females only make 1 viable gamete. They undergo meiosis twice still. They do make 4 cells, but 3 of them are going to be those polar bodies that aren't going to contribute to pregnancy. This is a very big difference between males and females. Males make a ton of sperm cells, 4 gametes every single time meiosis happens. Females will only make 1 gamete. That's the one that gets ovulated. Okay, and then lastly, the spermatid will undergo some maturation, will eventually form that spermatozoa, and you now have that sperm cell basically. Right? There's some additional maturation that has to take place in like the epididymis and all of that, but the point is you now have that classical sperm cell structure then. So where was all of this happening, by the way? All this mitosis and meiosis, like where in the male specifically was that happening? Yep. Absolutely. It's the seminiferous tubules within the testes, right? So like if you look at the testes, you have all of these, like they look like a big bunch of spaghetti. I think I just ruined spaghetti for myself. You have all these tubules, these seminiferous tubules within the testes where all of this is taking place. All of that mitosis with the spermatogonia and all the meiosis to make the spermatids and the spermatozoa, all within the seminiferous tubules. And keep in mind, this is what a tubule looks like, right? It's a tube And what you find is that the least mature sperm cells in spermatogonia are going to be found towards the outside of that tube, right, towards the edges of that tube. The more mature spermatozoa or the spermatids are all going to be found closer to the lumen, the whole of that tube, right? That's how we kind of distinguish the maturation phase. The further they get along that lumen, the closer they get to that lumen, the more mature they are. Okay, so that's kind of outlined nicely in this slide here, right? So this would be the kind of the wall of your seminiferous tubule, this white thing. This would be like the most immature sperm cells, your spermatogonia, and your spermatozoa would be way up here, closer to the lumen of the seminiferous tubule. Okay, so lastly, I just want to talk briefly about the hormones. Right, so we have a hormone, GnRH, gonadotropin releasing hormone, coming from the hypothalamus. It gets put into something, right? A special blood circulation. Does anyone remember what the name of that circulation was called that takes to the anterior pituitary? Yep. Not tract. Not, you're close. So hypothalamic hypophyseal something, but not tract. It's a blood vessel system. Keyword there is system. Portal system. Right? But good try though. And I'm making an emphasis on this because the hypothalamic hypophyseal tract is a separate structure, which I'll talk about in a second, right? So GnRH and any hypothalamic hormones like CRH, right? Corticotropin releasing hormone, GHRH. What else was there? Why am I blanking on this? TRH, that's the other one, and dopamine. Those are all made in the hypothalamus, and they get secreted into the hypothalamic hypophyseal portal system to act on the anterior pituitary. There's another structure called the hypothalamic hypophyseal tract, which is connecting the hypothalamus and the posterior pituitary. Okay, so keep that in mind. They're very similarly named to each other, but they are distinct structures. That tract is what's taking oxytocin and ADH from the hypothalamus to the posterior pituitary to be stored there. Okay, so don't make that mistake, right? Those are two separate structures from each other. And while I'm talking about this, let's bring in endocrine and try to tie things in together. Does anyone remember what kind of a hormone GnRH was? Yep. It's a neurohormone, absolutely. But what type of, like, is it a peptide neurohormone or a steroid hormone or an amine hormone? Any thoughts? Want to take a guess? Anyone? Not steroid. Not amine. Peptide. Okay, so every hormone released by the hypothalamus technically is a peptide hormone. Actually, no, not technically. That's what Dr. Bell said in her endocrine section. Technically, dopamine is an amine hormone, but I think in her slides or her PowerPoint, she says all hypothalamic hormones are peptide hormones, which I don't agree with. But anyways, the point is this is a peptide hormone. Okay, so start to make those connections. Where would you expect to find the receptor for GnRH? On the cell membrane or inside of the cell? In the cell membrane. Why? So it's a hydrophilic hormone, right? That means it can go into the blood and be perfectly happy interacting in the blood, right? It's a hydrophilic hormone. Blood is mostly water. Because it's hydrophilic, it cannot pass through the plasma membrane of your cells. That means the receptor has to be outside the cell. So it's on the plasma membrane, sticking out of the plasma membrane. That GnRH will bind to the cells in the anterior pituitary, and that then leads to the release of LH and FSH, gonadotropins. These are also peptide hormones. Okay, so these will travel in the blood. They're going to go through everywhere in the body, but there are specific cells within the testes that have the receptors for these, right? They're the target cells because they have the receptors. The same idea. The receptors are going to be on the plasma membrane, right? Because they're hydrophilic hormones. That also means they're perfectly happy traveling in the blood just fine. They don't need to be bound to any protein, right? So what exactly do these hormones do? Oh, I guess I don't have it on the PowerPoint slide here, but FSH is going to act on your Sertoli cells. LH acts on Leydig cells. Okay, so the way to remember that is L for L, LH for Leydig cells. Okay, FSH will act on the other cells, these Sertoli cells. What do they do specifically? LH promotes testosterone production and release from the Leydig cells. FSH promotes spermatogenesis on the Sertoli cells. Okay, in fact, Sertoli cells need FSH and testosterone from the Leydig cells to have spermatogenesis take place. Okay, so keep those things in mind. There's a lot of interactions taking place here. Now, of course, there's a lot of details I'm missing out, like the whole negative feedback system, but I think you guys have a good grasp on that. But do you have any questions on any of this or spermatogenesis before I move on? Are we doing okay? Is pacing okay? Have I lost anyone? I take the silence as a yes. Do you have a question? You're doing okay? Okay. I'm gonna keep going unless anyone wants me to stop and like cover anything else. Okay. Let's now talk about oogenesis. Okay, so same idea, and we'll tie in folliculogenesis with it as well. Okay, so oogenesis. A lot of similarities to spermatogenesis, but some very big differences, which we'll talk about here. Okay, we'll start off with the egg stem cells, which we call oogonia. Just like how we had spermatogonia, we have oogonia. Same idea, these are all diploid. Okay, these are all diploid. That means they have 23 pairs of chromosomes. Again, why do we say pairs? Because you have one pair from your dad. I shouldn't say one pair, one set of the 23 from your dad, and one set, the other 23 from your mom. 23 from dad, 23 from mom, come together to give you your 46. Every one of our cells has those 46 chromosomes, including those oogonia. Okay, but of course, if we want this daughter, this woman to give birth to a child, you want their chromosomes to be split off and just have 23 from her ovum and 23 pairs, or rather 23 from the sperm cells to come together, right? So you have to separate out those genes again, just like with spermatogenesis, which we'll talk about here. Okay, but again, keep in mind, oogonia have 23 pairs of chromosomes, just like every other cell of our body. Before birth, so this is big distinction number one compared to biological males. Before birth, all of these oogonia will undergo tons and tons and tons of mitosis. Okay, so when it came to the male, mitosis was taking place once puberty started. With the female, mitosis is happening before she's even born, while she's still in her mother, developing as an embryo. Okay, all those oogonia will undergo several rounds of mitosis. So everything we talked about before still applies. All 46 of those chromosomes will duplicate. Okay, again, we still call it 46 chromosomes, but they each have 2 of those arms, 2 chromatids, right? So 92 total chromatids. And then they will separate out those chromatids into genetically unique cells. Again, one oogonia turning into 2 oogonia that are completely identical to the cell that they started from. Okay, this is gonna happen several times. Okay, so the baby girl, while she's still developing, is gonna have millions and millions and millions of these oogonia before she's even born. And then before she's born, all of those oogonia will start meiosis 1. And at that point, we refer to them as primary oocytes. Okay, so what exactly happens? They start meiosis, meaning they cannot do mitosis anymore. This is it. And again, this is a big distinction between males. Males still have mitosis taking place, right, with the spermatogonia. But females, every single one of those oogonia will start meiosis 1. Okay, they're all going to start this developmental process to form that mature egg, although not all of them will make it there. Okay, so what exactly happens? Same idea. All of those chromosomes are going to get duplicated again. 46 chromosomes still, but each has 2 chromatids. Okay, so 92 arms, 92 chromatids for all those 46 chromosomes. Same idea in the male. You have crossover taking place, right? So genetic material from this baby girl who hasn't been born yet, from her parents, are going to be crossing over, right? So material from chromosome 1 is going to swap with material from chromosome 1 from the mom and the dad's chromosomes. Does that kind of make sense so far? Okay, so again, this is all happening before birth. These are all primary oocytes. And then at this point, they stay like this. Okay, baby girl is born and they're still like this. They're all primary oocytes. And then puberty hits, and that's where things start up again. Okay, so every month, once per month, some of those oocytes will be recruited to undergo this process of maturation to eventually form that one egg that's going to get ovulated. In reality, we have around 30 to 50 of those primary oocytes that are going to be recruited to undergo maturation. Okay, but here's the key thing. Only one egg is going to get ovulated. Even though 30 to 50 are being recruited, only one will get ovulated. Okay, this is the one that's going to get ovulated right here. It's the one that completes meiosis 1 and forms that one secondary oocyte now. right? This is the ovum, basically. This is what's going, well, I shouldn't say this is the ovum. This is the egg cell within the ovum that's going to be ovulated out. Okay, so here's a big distinction here. Look at this. It completes meiosis one. Same idea with the primary spermatocyte to the secondary spermatocyte. You're going to separate out dad's chromosomes from mom's chromosomes here into two distinct cells. One cell is going to be that secondary oocyte. The other one, though, is the garbage can for extra DNA. Okay, what we call the polar body. Again, when it comes to the female, she only ovulates one egg. Right, so where does that extra genetic material go? It goes into this polar body, basically just a garbage can for that extra set of DNA that's not going into that secondary oocyte. Do we have any questions on any of this so far? Okay. Now, I'm going to point your attention to something. A lot is happening between this and this. Okay? I know it says it's completing meiosis one. Keep in mind, there's a lot of folliculogenesis events taking place throughout this process right now between the primary oocyte finishing meiosis one and forming that secondary oocyte, right? So here's the slide here. Now looking at the follicle in relation to the oocyte, right? So again, remember, before birth, you have those oogonia. They did mitosis several times, made millions of themselves, and they all started meiosis one. That is when we classify them as primary oocytes. Baby girl is born, nothing happens, and then puberty hits. That's when those 30 to 50 oocytes will be recruited, and only one of them is going to get ovulated. Okay, for most of the time before ovulation, that primary oocyte is still a primary oocyte. It hasn't finished meiosis one yet. But the follicle, the support cells that are surrounding that oocyte, are developing throughout this whole process. This is folliculogenesis. Right, so this is where we talk about the primordial follicle and primary and secondary follicle and the Graafian follicle. These are the support cells that are supporting the oocyte, right? The granulosa cells and the theca cells, right? So at puberty, what happens is some of those 30 to 50 oocytes that get recruited go from being a primordial follicle, a very immature follicle, to form your primary follicle. They will continue to mature to form your secondary follicle and continue to mature again, Graafian follicle. There's only going to be one dominant Graafian follicle, even though we've recruited 30 to 50 eggs, one dominant Graafian follicle. That is the one that's going to finish meiosis one to form the secondary oocyte, and that is the one that's going to be ovulated. Okay, so that is all happening in this early to mid follicular phase, right? You have the development of the follicle, right? So that primary oocyte is still a primary oocyte, but it's the follicle that's going to be developing. Right, so let's look at this in kind of detail here. Right, so all eggs in a female will be primordial follicles, but every month, 30 to 50 of them will be recruited, and they will go on to mature, and only one of them is going to form that dominant Graafian follicle. So what are the differences here? Well, primordial follicle has that primary oocyte, that egg cell that's still begun meiosis one, but has not finished it, and it has some very immature granulosa cells. 30 to 50 of these are going to get recruited, and they're going to form your primary follicles. Okay, so again, you still have a primary oocyte. You still have those granulosa cells, which are now yellow. They're starting to mature and starting to make all sorts of hormones, like estrogen, for example. And you have a new structure called the zona pellucida. And Dr. Bell put it really nicely. This is basically the egg shell, right? The shell that's surrounding the oocyte that the sperm cell has to bind to. Some of those primary follicles are going to go on to form secondary follicles. Right, so more maturation taking place. You can see the granulosa cells have really started to grow. There's way more of them. You start to see your theca cells starting to grow at the very outside. Right, this is how you distinguish the secondary follicle from the primary follicle. That those theca cells being present and way more granulosa cells. Okay, so we started with 30 to 50 getting recruited. Maybe some of them made it to this stage, maybe like 10 or 15, I don't know. But only one of them is going to make it to the dominant Graafian follicle. It's the same idea, way more granulosa cells, larger, more mature theca cells. You still have the zona pellucida, and now you have this structure called the antrum, basically a fluid-filled structure that's going to help with ovulation. Creates this kind of like hydrostatic pressure that's going to like literally catapult the ovum out of the ovary. Okay, so again, only one dominant Graafian follicle is going to be formed. This is the one that's going to have the oocyte that finishes meiosis one and starts to form the secondary oocyte. Is that all good so far? That is where you have the separation then of dad's chromosomes from mom's chromosomes at that point. Okay, so let's go back to this slide here, if I can find my cursor. Right, so that's happening. All those folliculogenesis events are all happening before this meiosis 1 event takes place. And again, keep in mind, not all egg cells are doing this. Only 30 to 50 are being recruited per month. Because if all of them are recruited per month, that means a woman would have one chance, 1 month basically, to get pregnant in her whole life. Okay, in reality, this happens from puberty up until menopause. So there's several chances for that individual to get pregnant. Okay, so question for you. Does anyone know what specifically is recruiting those 30 to 50 follicles to undergo maturation and go through this whole process? Yep. FSH specifically, absolutely. So in your early to mid follicular phase, you see that you have relatively high FSH levels. That's this kind of blue line right over here. This is the hormone that's gonna be put into the blood, gonna circulate throughout the whole body, but it's gonna bind to receptors specifically found on these follicles. Okay, that is what's gonna lead to the recruitment of those follicles to start to undergo maturation and develop to form those more mature follicles. Okay, so FSH is what leads to the development of the follicle. FSH is also what promotes estrogen production, right? So where's estrogen specifically coming from? It's not the oocyte. There's 2 support cells here. There's granulosa cells and theca cells. What is estrogen specifically coming from? You have a 1 in 2 chance. I'm going to pick on someone. Any thoughts? Dryden, I know you. Where's it coming from? Granulosa cells or theca cells? You had a 1 in 2 chance. Those granulosa cells, it's all good. Okay, estrogen is coming from your granulosa cells specifically. And I'm going to show you what that looks like in 2 PowerPoint slides here. Okay, this estrogen, this rise in estrogen is coming from the granulosa cells. Now you'll notice in the early to mid follicular phase that FSH is going down, right? It starts off relatively high and you can see it's progressively going down. Why is it going down specifically? Think about the negative feedback system. If estrogen levels are rising, do you want to keep making FSH? Right? Your classic negative feedback system is when you're making a lot of hormones and levels get high, they're going to act back on, the hypothalamus or the anterior pituitary and suppress the release of the hormones that made that hormone in the first place. Right? So I'm going to show you what that looks like now. Estrogen, as it's rising, is going to act back on the hypothalamus and suppress GnRH, and the anterior pituitary to suppress LH and FSH. Okay, so initially in that early to mid follicular phase, it has a negative feedback role in preventing the release of more GnRH, LH, and FSH. And if you look at the graph, you can see that nicely, right? You can see FSH specifically going down. LH was already low to begin with, so it doesn't really get affected by this, but FSH is going down. So here's something interesting. If FSH was needed to bind to these cells and lead to the production of estrogen, how is estrogen still continuing to rise if FSH is going down? And the answer is the estrogen is coming from these granulosa cells. Look how many cells there are, especially in this large dominant Graafian follicle. Even though FSH is going down, whatever amounts are in the blood, there's still going to be small amounts there, are still going to bind to these granulosa cells and promote estrogen production, right? So this is how you still get a rise in estrogen, because you have so many granulosa cells. But this is also why some eggs just do not make it to this stage, right? You only have 1 dominant Graafian follicle, but if there isn't enough FSH for all the eggs, the weaker ones are going to die, and you end up with 1 mature dominant Graafian follicle. This is the sort of mechanism that leads to that. Eventually, though, something really cool happens. At this stage over here, it's called the mid to late follicular phase. Estrogen levels kind of hit this threshold value, right? This magical number, and all of a sudden, FSH and LH levels skyrocket, right? So what exactly is happening here? Well, this is having to do now with a positive feedback system, right? So for some reason, as estrogen levels get really, really high for some period of time, they'll actually act on a distinct set of cells within the hypothalamus and the anterior pituitary and cause more release of GnRH, LH, and FSH. It acts in a positive feedback way instead. And that's what gives rise to the ridiculously high levels of LH and FSH. The LH specifically, that LH surge, what does that end up doing? Yep. Absolutely, right? That's what leads to ovulation, right? So that huge amount of LH right up here, that is the trigger for ovulation, right? So right before this, that primary oocyte has finished meiosis 1. It's formed the secondary oocyte, and now you have this LH surge, and that oocyte gets shot out of the ovary and into the fallopian tube and is now an ovum. A secondary oocyte that we now classify as the ovum now. Any questions on that so far? Okay. While I'm talking about the hormones, I'll draw your attention to like FSH and LH again, right? So FSH was acting on the granulosa cells and leading to the production of estrogen. LH, this slide is a little bit messed up. LH is acting on your theca cells to lead to the production of this androgen, androstenedione. So remember, when it came to the males, LH acted on Leydig cells. What did that cause? It led to the production of testosterone, right? Same idea here. LH, and again, messed up here. LH should be here. LH is going to act on now theca cells. So these are equivalent to the Leydig cells to lead to the production of an androgen, right? Just like testosterone, except this one is called androstenedione. This, though, can then be shuttled to your granulosa cells, and they have a special enzyme that can convert that into estrogen. That is how granulosa cells make estrogen. They can literally convert that androgen into estrogen. And that is why you see that rise in estrogen during the early to mid follicular phase and the late follicular phase as well. Okay, then ovulation happens, right? So that LH surge gives rise to ovulation. And at this point now, that primary oocyte has already finished meiosis 1, right? So this secondary oocyte, that is what's going to get ovulated out with some granulosa cells. That's what forms that ovum, tying everything back together now, right? And again, keep in mind, you have to have that polar body, which is sort of the garbage can for the extra DNA, right? The extra genetic material that you do not want to have in that secondary oocyte. Now, here's a big distinction between sperm cells and egg cells. Secondary oocytes are just started meiosis 2 at this point. They have not finished meiosis 2. Unlike sperm cells, which have finished meiosis 1 and 2. What has to happen to the secondary oocyte for meiosis 2 to finish specifically? Yep. Absolutely. Fertilization, right? So again, that's a big distinction right there, right? And I can see Dr. Bell asking you a question on the difference between spermatogenesis and oogenesis. Here is another big one. Secondary oocytes have not finished secondary or meiosis 2 until fertilization has taken place. Once the sperm cell has bound to the egg, it's kind of gone through the zona pellucida, it's broken its way through, and it's entered into the cytoplasm. That is then when that secondary oocyte will then finish meiosis 2. Right, so you pull apart those the double genetic material, right? Those two chromatids into, again, one, now the zygote eventually is what it's going to be called. And again, that extra genetic material is going to be put into another polar body, right? So that way you have, again, single 23 strands of chromosomes that can then combine with the 23 single strands of chromosomes from the sperm. And now you have your 23 pairs of chromosomes, right? 23 from dad, 23 from mom, for the total of 46. That then gives rise to the embryo, right? That's what we are now referred to as the zygote. You're now diploid at that point. Any questions on that? Okay. Okay, so again, a lot happening in that step right over there, right? Fertilization, a lot of things that have to happen, right? So the ovum is not just a secondary oocyte. It has some cells still attached to it, right? Some of those granulosa cells went along with it when it got ovulated. We call those cumulus cells now. Okay, so those are kind of like the support cells surrounding and kind of nourishing the secondary oocyte. And again, keep in mind, the egg still has its egg shell, right? That zona pellucida, right? You can kind of see this, like, it doesn't really show too well on the PowerPoint slide here, but I can see a really nice little layer here just outside the ovum. Right, so for the sperm cell, for it to deposit its DNA into the egg, it has to make its way through the cumulus cells. It's going to literally break them apart using an enzyme. It's going to reach that egg shell, the zona pellucida, and it's going to bind to it. And at that point, it's going to literally break its way through that egg shell. Right? That's what's referred to as the acrosome reaction. Right? So remember, the sperm cell has a specific structure in its head called the acrosome. Okay, it contains all of these enzymes that can literally break apart the zona pellucida and allow the sperm to enter into the egg. And then at that point, the plasma membrane of the sperm and plasma membrane of the egg can fuse together, and now you can have the genetic material coming together. And you also have this cortical reaction to prevent other sperm from fertilizing that egg once one sperm has done it. Okay, so that's the cortical reaction to block multiple sperm from fertilizing that egg further, right? Again, you do not want to have too many copies of chromosomes. You want to have single 23 strands of chromosomes from the sperm, 23 single strands of chromosomes from the egg to get you your 46 chromosomes. Okay, so the sperm head, enters the cytoplasm. That is then when meiosis 2 will complete. And then the nuclei, the 23 chromosomes for dad, 23 chromosomes for mom come together. And that is then what leads to the zygote. Okay, so that was oogenesis, folliculogenesis, and fertilization in a nutshell. Do I have any questions on any of that before we talk about what's happening to the uterus and implantation and all that? Have I lost anyone? Be honest. I know it's a ton of information on a Tuesday morning. It's still 4 days. You haven't really studied, like you haven't crammed like full on yet. But again, if you can follow what I'm saying, that's the important thing, right? You can always study for the minor details later on. It's more of understanding the material from now so that it's easier to remember those things. Okay, so now we could talk about what's happening once the egg has been fertilized, right? So this idea of now pregnancy and implantation and the developing embryo. At this point, we're now past ovulation, right? Ovulation happened at day 14 of the, oh wow, that's really messed up. Day 14 of the 28-day cycle. We're now in the luteal phase, right? So back in the ovary, there are still some cells left behind that did not get ovulated. They form that structure, the corpus luteum. Now, instead of making a ton of estrogen, this structure is going to make a lot of progesterone, right? Now, here's something interesting. You'll notice that at the end of the cycle, progesterone levels really kind of plummet, right? Because of that, that's caused by the corpus luteum kind of degrading away and dying off. What is this drop in progesterone going to cause? What's going to happen on day 1 of this whole cycle? Yep, absolutely, menses, right? Menstruation is going to take place. So the corpus luteum dying means that progesterone levels are going to plummet, and that drop in progesterone is what triggers menses or menstruation, right? So the shedding of the uterine lining. Can an implantation happen if that were to occur? No, right? The baby, the zygote has to implant in the endometrium, and it can't do that if there is no endometrium. So during pregnancy, we have to have a way in preventing this corpus luteum from degrading, right? Because this is what's leading to the progesterone release. And we do not want that progesterone to drop if we want implantation to take place, right? So we're going to talk about what specifically happens to prevent that from happening shortly in just a second here. But keep in mind, there are other events taking place throughout this whole process, right? So we talked about day 1 to day 28 in regards to the oocyte and folliculogenesis. You can also refer to it as the uterine events, though, what's happening from day 1 to day 28 with regards to the uterus. Right, so there's day 1 right over there, right? The endometrium has been shed. That is menses, lasting for about 5, give or take 5 days. I'm sure it varies, but the point is, it's going to be shed as a result of that drop from progesterone. Why did we have a drop in progesterone? Because that corpus luteum dies, it degrades away. And that is the trigger for menstruation. Then you start to have the buildup of that endometrial lining again, right? So that's what we refer to as the proliferative phase. What specifically is contributing to this proliferative phase? What is causing the growth of this endometrial lining again? Because it's not progesterone. We just saw that it completely dropped off. What other hormone would be involved here? Estrogen, absolutely, right? So the hormone released by those granulosa cells, right? The follicle itself is being released, and that is what's leading to the growth of the endometrial lining, right? It's starting to grow back up again because of that estrogen. So we call that the proliferative phase because the cells are literally dividing, proliferating, undergoing mitosis, and forming layers and layers and layers. Then ovulation happens, and now we're not making estrogen anymore. We're making progesterone from that corpus luteum. right? The leftover cells that did not get ovulated during ovulation. This is now going to release progesterone. This now contributes to the secretory phase of the uterus, right? So instead of it growing a lot, it is still growing, but not as much as the proliferative phase. In this phase, though, we're secreting things into the endometrium, nutrients, blood vessels are growing, all sorts of like growth factors in the endometrium in preparation for an implantation to take place, right? So once that zygote implants its way in, it now has access to all those nutrients and can start to grow then, right? So the secretory phase is so important for making sure the endometrium is ready for the implantation to take place. And again, if there's no pregnancy, that corpus luteum is going to degrade away, progesterone levels are going to plummet, and that is then what leads to menstruation happening again. And now we're back to day one again, right? So that's the cycle happening every single month, over and over and over again. Does that make sense? Do we have any questions? Okay. So let's finish off with implantation now, right? So we have a fertilized zygote. It's going to start to divide several times, right? It's undergoing mitosis now. Obviously, we have to go from one single cell, right, the zygote, to the trillions of cells that make up our body. They have to form the brain, they have to form the heart, the kidney, the respiratory system, my pinky toe, my thumb, my eyelashes, who cares, right? It has to form all of those, right? So these cells are going to undergo rapid mitosis, right? So they go from one cell to two cells to four cells to 8, 16, 32, 64. Eventually, you form what's called a morula, basically just a ball of cells. This is where you start to see some really interesting structural differences. Then the next step is even more profound, where you have the blastocyst. This is around day 5 after fertilization has taken place. This is the cell, or rather the structure, that is going to implant into the endometrium. You start to see some really unique structures. On the outside, you have the trophoblast cells. And on the inside, you have the inner cells, inner cell mass. What is the inner cell mass going to give rise to ultimately? The little baby, right? This is what gives rise to the person. This is what's going to give rise to every single structure of the human body. The trophoblast cells are going to be the ones that actually implant into the endometrium, and they form the placenta, right? So they're going to form the structure that allows the maternal nutrients, the maternal blood, to give access to or allow exchange of nutrients and gases to the developing embryo, right? So the trophoblast cells do actually quite a bit. The inner cell mass is going to give rise to the baby. Trophoblast cells are very important for allowing implantation and then supporting the actual embryo itself. Okay, so again, keep in mind, this is all contingent on that endometrium being present, right? You do not want it to be shed away during menstruation. So if we want to prevent that from happening, you have to prevent the corpus luteum from degrading. How do we do that? Well, the placenta is going to release a hormone called, anyone? The hormone that the placenta releases that prevents the corpus luteum from degrading. H. Not LH. CH something. HCG. HCG. Okay, so human chorionic gonadotropin. Okay, so that is going to get released by the placenta. That's going to go throughout the whole blood, and it's actually going to act back on the corpus luteum and keep it alive. What does that do? It's going to lead to progesterone levels being sustained. Right? The corpus luteum is going to continue to make progesterone so that the endometrium is not triggered to shed. Because remember, we already talked about this. Once progesterone levels drop, that's what triggers menses. Right? You no longer have the endometrial lining. Without the endometrial lining, you do not have anywhere for the baby to develop and grow. There's no place to implant. Right? So the HCG, human chorionic gonadotropin, is what preserves the corpus luteum and keeps it alive. Does that make sense? Okay, awesome. Okay, so again, inner cell mass, that's what gives rise to the baby. Trophoblast cells do a lot. They're going to actually attach to the endometrium. They're going to digest up the endometrium so that the embryo can actually get into the endometrium where all those nutrients were, right? Remember, the secretory phase had the blood vessels and all the nutrients. There's probably a ton of glycogen there. I'm going to tie all that together with digestive. That's going to give you access to the baby to all those nutrients. The trophoblast cells will also break apart the blood vessels so that the baby now has direct access to the maternal circulation with all her hormones and nutrients and gases and everything that it needs to survive. Okay, and they developed something called lacunae. Basically, they're going to break these blood vessels and form pools of blood that now give perfect access to all those things that we just talked about. Okay, so again, trophoblast cells do a lot. And I can see Dr. Bell asking a question on what trophoblast cells do specifically, right? So there's kind of three main things. Attaches to the endometrium, digests the endometrium and creates the lacunae, and it's also going to form the placenta. Okay, so that is the lifeline that connects the maternal circulation and the fetal circulation. Okay, you never have blood directly attached to each other, but it provides that area for exchange of nutrients and hormones and growth factors. Okay, and also the placenta, I just said, is what's releasing HCG, right? So technically the trophoblast cells, well, they're the ones that are going to form the placenta, which are the ones that make HCG, right? So trophoblast cells, the main point I'm trying to make here is they do a lot just beyond attaching to the endometrium. Okay, and then lastly, you now have the baby developing, right? So you can see those lacunae, these like pools of blood from the broken blood vessels. And now the baby makes these structures called chorionic villi, right? These kind of like tree-like structures that kind of reach out into these lacunae. And the blood vessels of the baby that are kind of going to attach to the umbilical cord are found within these chorionic villi. This is where exchange can take place now, right? So oxygen can come in, carbon dioxide can come out, nutrients can come in, waste products can come out, and so on and so forth. Right? That's all going to happen at the placenta. And again, keep in mind, this is what's going to lead to HCG being produced, right? The placenta, right? So HCG, that is what pregnancy tests are basically looking for, right? That is what tells you you are pregnant, because that is coming from the placenta. It's coming from the embryo. It's like the extra embryonic structure itself, right? So that's a good way to tell you if you are pregnant. Again, what does HCG do specifically beyond telling you you're pregnant? Well, it's going to signal to that corpus luteum and tell it to stay alive. Okay, it's going to prevent it from degrading and keep it to make more progesterone. Right, that progesterone is so important for preventing the endometrium from being shed. You don't want menstruation happening now, right? The embryo is there, it's implanted. You don't want that endometrium to be shed away, and now the baby is gone as well. Right, so keep in mind, HCG is very, very high, especially early on, and that is what maintains high progesterone levels. Eventually, they drop off because the embryo itself, like the placenta rather, will take over the corpus luteum and make its own progesterone. And that is then what will keep the endometrial lining maintained. Okay, so you no longer need that corpus luteum then after that. Okay, that is my overview of reproductive physiology. Again, I didn't cover everything. Again, you're required to know everything, even though I didn't cover it here. So just keep that in mind. But I think I covered the main topics there. Okay, so do we have any questions on reproductive phys? Okay, we're into the last hour now. Now we can move on to everything else. Okay, so again, keep in mind, only 30% of your questions are going to be related to everything outside of digestive and reproductive. Okay, so I've picked topics that I think are relatively important. So an excitable cell, graded potentials versus action potentials, and the events at the chemical synapse. Again, that does not mean the other things that Dr. Baya taught are not important. I can totally see her asking a question on tonicity, right? So remember, hypertonic versus hypotonic versus isotonic. I can see her asking a question on that, even though I have not listed it here. Okay, so keep studying for those things. That's a very important topic. But I think that graded potentials and action potentials tend to be one of those topics that most students have trouble with. So that's why I picked it for today. So again, I'm going to leave it up to you guys. Is there anything anyone would like me to cover specifically in any of the older units? Otherwise, we'll just go through these basically one by one and see how much we can get through between now and 1 o'clock. And I also have a bunch of things from motor. I am skipping autonomic nervous system and muscle because I think there were like 2 or 3 questions between both of these. So it's not even worth our time to spend time on it. And I spent a little bit more time on cardiovascular, renal, and then respiratory especially. Okay, should we just go for it? We'll start off with graded potentials and action potentials. Okay, that's not it. Hang on. Do we need to take a break or are we okay to keep going? take the silence as a yes. All right, who remembers this? Oh, no, indeed. Okay. Again, when you're studying for these things, these pop up a couple times throughout the course, right? These come up in sensory and motor. Action potentials come up in cardiovascular as well. They come up in muscle physiology and autonomic nervous system. These are all very interconnected with each other. Don't look at your workbook as holy crap, a crap load of information that I have to memorize. Make those connections because that's going to make it easy to remember. And you're going to realize there's actually way less to remember than you think because there's so much overlap between a lot of these concepts. Okay, so keep that in mind. Graded potentials versus action potentials. In a very simple sentence, how do we differ between these two? Simply put, action potentials are the way that our body signals. Okay, so with regards to neurons, it's the way that our nervous system communicated with itself. With regards to the heart, it was the way that the heart cells contracted. With regards to the hypothalamus and the anterior pituitary and the posterior pituitary, action potentials is what led to the release of those neurohormones back in the hypothalamus and the posterior pituitary, right? It's the way that our body signals to other parts of our body. It could be involved in long distance communication. A graded potential is a way to decide whether or not an action potential is going to get fired. Okay, so I'm going to say that again. A graded potential is how our body decides whether or not it's worth it to fire an action potential. Okay, because keep in mind, action potentials, there's a lot going on. You can imagine there's a lot of energy needed every time you fire an action potential, right? So you don't want to constantly fire action potentials because A, it would drive you nuts over time if you have a lot of information coming at you and there's so much happening. So graded potentials are a way to decide between what we consider as important stimuli versus less important stimuli, right? So the example that Dr. Baya gave way back in September was a light touch versus a stronger, heavier touch. Okay, so a light touch is still going to activate the receptors in your arm. Let's say something was brushing against my arm, right? Tying in Dr. Everling's stuff, you're going to have mechanoreceptors in your skin. Remember Meissner's corpuscle and Pacinian corpuscle and all that stuff? Yeah, all that good magic. That's going to activate those receptors. But a light touch, maybe it's like a small bug on my arm, is not necessarily all that relevant to send that information to my brain. It's not worth the energy expenditure for that to occur, right? So a graded potential is going to take place, right? So you still have signaling happening, right? You have your receptors getting activated that leads to, say, an opening of ion channels, right? If it's mechanoreceptors, that means you have mechanically gated stretch channels in your arm being activated. That's going to open up ion channels and allow some sodium to come in. So you have a little bit of depolarization, right? So the influx of positive charge. But here's the thing. By the time it gets to that trigger zone, remember the axon hillock? It's down below that threshold value, right? We have to have that value of minus 55 millivolts by the axon hillock. The reality is, if something is so light that's touching my arm, it's not allowing enough ion channels to open, meaning there's not going to be that activation of threshold. Are we going to fire an action potential in this situation? No, right? That is how our body differentiates between the important versus the less important stimuli, right? So if, say, for example, someone was punching my arm, well, that's way more important than a light bug kind of crawling on my arm, right? So what happens? More receptors are going to get activated, more of those mechanoreceptors, probably some pain receptors too. You're going to have way more ion channels opening up and way more influx of positive charge, right? So a lot more sodium coming in. Now, even though you have ions moving down, remember you have decay as well, right? Ions leaving the cell. Regardless, because there is such a high influx of positive charge, by the time you hit the axon hillock, you're going to hit threshold. Are you going to fire an action potential? Yes, right? You've hit that threshold value by the axon hillock. What is the consequence of that? I felt the punch, right? That information made it all the way up to my brain. That information signaled to my brain and told me something touched my arm. Okay, that is what the purpose of an action potential is. It's signaling, it's communication, it's telling my body what is going on, right? So that is how you differ graded potentials versus action potentials. Does that make sense? Sort of, kind of? Okay. Let's get into some more specifics again, right? So this is the action potential. Remember this was like our first activity together in tutorials, right? So keep in mind, most neurons have a resting membrane potential of around minus 70 millivolts. That is what it's at rest. Then a graded potential happens, right? Maybe something touches your arm, or another neuron is signaling to another neuron and giving it a excitatory postsynaptic potential. That's going to lead to the influx of positive charge. If that is enough to hit that threshold value at the axon hillock, that minus 55 millivolts, you're going to have an action potential happening, right? So depolarization, repolarization, and then hyperpolarization. Remember all those things? Right, so the graded potential in this diagram is happening way over here, right? This kind of left part over here. That is what gives rise to you potentially hitting threshold. Okay, the action potential is specifically once you've hit that threshold value, once you've hit that minus 55 millivolts. The depolarization, repolarization, and hyperpolarization are part of the action potential then. Okay, so here's some additional differences between graded potentials and action potentials. Okay, so again, action potentials are all or none. You either hit that threshold value and have the action potential, or you do not hit that threshold value and nothing happens. Versus graded potentials can happen to any degree that they can, right? As long as something is activating it, ions are moving in, that is a graded potential. Action potentials are the same amplitude for a given cell, right? So in this example, you can see that this neuron is hitting plus 30 millivolts. By the way, I'm going to guess you do not have to memorize those numbers for the exam. That's something that Dr. Baya kind of emphasized. The point is, an action potential will always hit that value. A graded potential can differ, right? Think about the light touch versus the strong touch or heavy touch, or the punch, for example, right? The light touch is going to have a very low amplitude graded potential. A punch is going to have a way stronger amplitude graded potential, right? That's what's going to lead to way more ions coming in, a way higher amplitude of influx of charge. Graded potentials are going to go down very specific kinds of ion channels. Anyone remember the types? It's okay if you don't. It's been long enough. Right? So sodium, absolutely, but I'm taking a step even further back than that, right? So when it came to action potentials, we're relying on voltage-gated channels. When it comes to graded potentials, what is the gated thing there? Ligand gated, right? So chemically gated, absolutely, and anything else? Mechanically gated, absolutely, right? So for example, if something is touching my arm, that is a mechanical stimulation, right? That is physically deforming the membrane of those cells. That is mechanically gated channels. Chemically gated, for example, taste or smell. Those are chemicals that are literally moving onto my tongue or into my nose and binding to receptors. Those are going to open other sets of ion channels and allow influx of ions to come in. Okay, so that's a big difference. Graded potentials are mechanically and chemically gated channels. Action potentials are voltage gated. Why are they voltage gated? What does that mean even? Like, what does it mean to be gated by a voltage? Go ahead. Exactly, right? So the main thing I'm trying to emphasize to everyone is don't memorize stuff like that, right? It's in the name. It's a voltage gated channel, right? What would you say to a coin operated or a coin gated channel? You'd have to put a coin in there for it to open, right? It's like a parking lot where you have to put in like money for the gate to open up, right? Same idea here. It's gated by a voltage. Well, guess what? We are affecting voltages throughout here, right? When we're hitting threshold, that is a voltage change. Threshold was the gate that's going to open up those voltage channels, that minus 55 millivolts, right? So that's why in action potentials, we're relying on voltage gated channels. And keep in mind, action potentials will only travel in one direction, right? From that axon hillock all the way down. We're going to talk about why in just a second here. Graded potentials kind of go dispersed everywhere. They kind of move around everywhere. And that's what leads to decay, right? Ions are literally moving and leaving the cell. That's what leads to ions leaving and causing that decay and that charge then, right? So the values, the voltage actually ends up declining over time. Okay, so graded potentials. Again, the stimulus is either going to be a mechanical stimulus, something touching my arm, for example, or it could be sound waves, right? Remember the whole sound part of the sensory unit? Sound waves coming into my ear and activating the hair cells within my cochlea, right? Those are going to bend the hair cells. Those are mechanically gated channels. Chemically gated channels for taste or smell. Those are chemicals literally binding to receptors in my nose or in my mouth and activating them. right? And especially if they're activating them and information is reaching my brain, they're probably depolarizing. Right? It's probably going to lead to an action potential. It's probably going to be a positive charge, right? So sodium is primarily going to be the trigger for that. But graded potentials can be hyperpolarizing as well, right? This is what leads to inhibitory postsynaptic potentials, right? So thinking back to the motor section when we talked about reflexes, right? That hitting the knee reflex where your knee stretches out, there's two things that happens there. First, your leg is going to extend out, right? So the leg is literally stretching, but you're also going to inhibit the flexor muscle, the antagonistic muscle, right? So what happens is you have an excitatory postsynaptic potential that stretches the leg out, and an inhibitory postsynaptic potential on the muscle that prevents it from stretching out, or rather from flexing, right? So preventing that stretch from taking place, right? So that's another big distinction. Graded potentials can be inhibitory. Right, so that's what gives rise to your EPSPs and your IPSPs. Depolarizing graded potentials are excitatory. They promote action potentials, right? So primarily that's going to happen by sodium entering in. That is how you get depolarization. That's how you hit threshold. Hyperpolarization happens by potassium leaving, right? If you open up potassium channels, where does potassium want to go? Where is potassium located higher? Inside of cells, right? That's sodium or salty banana idea again. Chloride is higher outside of cells. So if you open up chloride channels, it's going to come in, but chloride is a negatively charged ion. It's going to reduce the voltage within the cell. Okay, so that's going to inhibit the cell and prevent it from firing an action potential. Okay, another distinction, location. Graded potentials are happening where specifically? In a neuron. Yep. Absolutely, right? So the soma, which is literally the cell body of the neuron, and the dendrites, which are these like projections sticking out of it, right? So that's what we refer to as the input zone. Action potentials are specifically happening from the axon hillock and down the axon to the axon terminal. Okay, so a big distinction there is the location between the two. Channels we talked about, graded potentials are mechanically and chemically gated channels. Action potentials are relying on voltage gated channels. Okay, so let's talk about the action potential. This was, I think, our second or third activity together in tutorials, right? So the depolarization phase. Does anyone remember what specifically is contributing to this? What ion is leading to this depolarization? Absolutely, right? So sodium is going to enter into the cell. And remember, are these chemically gated or mechanically gated or voltage gated channels at this point? Not chemically, because we're talking about an action potential now, right? Voltage gated, absolutely, right? So in the depolarization phase, you have sodium voltage gated channels opening up. What was the trigger for them to open? Well, they're voltage gated, right? That means they were triggered to open by a voltage. What specific voltage? Threshold, right? That minus 55 millivolts. That's what opened up that channel. Now, again, I don't think Dr. Baya is going to ask you too much on the activation versus the inactivation gate, I hope, but I think it's useful to remember anyways, right? So your activation gate is going to open up and allow sodium to enter in. Your inactivation gate was already open to begin with. So you have a fully opened up channel. Sodium is going to come in, right? Because sodium is higher outside of cells compared to the inside. Sodium floods in. What happens to the voltage inside the cells? It's going to skyrocket. It's going to become more positive. That is what depolarization is, because sodium is a positively charged ion. Now, potassium voltage gated channels are technically also triggered to open here, right? They are also voltage gated and they are triggered by that minus 55 millivolts. Why are they not open here though, even though they're triggered to open? What was special about the potassium voltage gated channels? Yep. Absolutely. So these ones I won't say are leaky because you have separate potassium leak channels. These are gated channels. Okay, so they actually have a gate on them that prevents leak normally. Okay, but here's the key thing here. They are slow to open. Okay, so they are triggered by minus 55 millivolts, just like the voltage on the sodium voltage gated channel, but they are slow to open. So during this phase, go back to depolarization here. During this phase, a ton of sodium is flooding in and no potassium is leaving out. A lot of positive charge accumulating inside of the cell, and look at that. The voltage skyrockets. Right? That is what depolarization is all about. What is the next phase called after depolarization? Repolarization, thank you. And what is this primarily being driven by now? Depolarization with sodium coming in. What is repolarization driven by? Yep. Absolutely, right? So we said that the potassium voltage gated channels were slow to open in the previous stage. Now they are open. And where's potassium going to want to go? Well, potassium is higher inside of cells, right? So as soon as that gate opens up, potassium is going to flood outside of cells, right? Down its concentration gradient. What's going to happen to the voltage inside the cell? It's going to go down now because positive charge is leaving. Simultaneously, your sodium channel is going to close up now, right? So your inactivation gate specifically closes up. Can sodium come in now if this gate is closed? No, right? It's literally a doorway preventing sodium from moving through. So now sodium cannot come in and potassium can leave. What happens to the charge inside the cell? It's going to now plummet, right? Because you literally have positive charge leaving. And keep in mind, you still have leak channels, right? So sodium can still leak out of the cells as well. So you really have a lot of positive charge leaving the cell. Now, potassium was slow to open and it's slow to close, right? So once we've hit resting membrane potential again, that minus 70 millivolts, that potassium channel will want to close back up again, but it's slow to close. So the consequence is that the voltage actually dips below minus 70 millivolts. What do we call this stage of the action potential now? Hyperpolarization, right? Okay, so we're now even more polarized compared to resting membrane potential. Potassium will continue to leave because this gate is slow to close. At this point, the sodium channel is actually back to what it was at the beginning. Activation gate is back closed again, and inactivation gate is now open. So in theory, if you hit threshold again, you could open this up again. It's just very difficult to do because you're now hyperpolarized. You're even lower, further away from threshold now. You're more negative because all that potassium is leaving. Okay, then potassium will stop leaving eventually because that gate will finally close, right? So sodium can't come in, potassium can't leave, and now you're back to resting again, ready to fire another action potential. Any questions on that? Did that bring back good memories? Okay, good answer. So how does this relate now to the, oh, actually, let's talk about this first. Yeah, so I kind of briefly mentioned that action potentials will travel in one direction, from the axon hillock to the axon terminal. Why? What is preventing that sodium that's coming into the cell from going backwards up towards the dendrites? Exactly. So here's what's happening. As the threshold is reached and those voltage gated channels are opening, sodium is flooding in. Sodium wants to come in because the inside of the cell is negative, right? Sodium is a positively charged ion. It wants to come in. Initially, it's only going to open right where threshold was reached at the axon hillock. Then sodium is going to see ahead of it, all this like further part down the axon is all still negative, right? So sodium wants to move forward towards those more negative regions. What happens? Those regions hit threshold. They go up to minus 55 millivolts, and now they open up their voltage gated channels, right? So you can see that here in the second part of the diagram, and it continues on and on and on. This action potential literally moves from one segment of the axon down to the further segments of the axon, right? Because it's all negatively charged ahead of it there. Now, here's the thing. Once sodium is down towards this part of the axon, you'll notice that this part of the axon actually has some negative charges associated with it again, right? Because now it's in your repolarization or potentially in your hyperpolarization phase. So technically, sodium could go backwards, but an action potential will not be triggered. Why? Because back in the repolarization phase, that inactivation gate is still closed, right? So even if you hit threshold again, that's not going to open that inactivation gate, which means sodium cannot flood back in again. You cannot have another action potential happening, and that means an action potential can only go in one direction. Does that make sense? I can see that being a question on the exam. Dr. Baya asking, what is the, what contributes to the unidirectional movement of sodium down the axon? The answer would be your sodium inactivation gate. Or yeah, the action potential not going backwards, basically. Okay, any questions on that? Did I miss anything on that slide there? Hello, sodium voltage gate, that's fine. Okay, that's fine. Okay. Okay, let's finish off with your chemical synapse now. Okay, so all of this is happening, that action potential is traveling down the axon, and eventually it's going to reach the very, very end of the axon. Again, remind me, what is that area called at the very, very end of the axon? Axon terminal, right? That is the very, very end point. What happens when the action potential gets there? Well, presumably there's something else beyond that cell. There might be another neuron, or if it's the muscle unit, this could be your alpha motor neuron and your actual skeletal muscle cell just below it, right? So there's some sort of signaling that has to take place from one neuron to the next cell, whether it's another neuron or a muscle cell, for example. So in this case, we call this a chemical synapse. Synapse is what we call the gap in between, and we're relying on chemicals to transmit information from the first neuron to the next cell. Okay, so we have a presynaptic neuron here and a postsynaptic neuron after that synapse. Okay, so here is that zoom in here, right? So you can see the axon terminal of that presynaptic neuron. It has all these vesicles with the neurotransmitters in it. When the action potential reaches all the way down to this axon terminal, that is going to activate another set of voltage-gated channels. Like these are calcium voltage-gated channels. Where does calcium want to go? Inside the cell or outside of the cell? Inside of the cell, right? Calcium is higher outside, right? So when you open up these channels, calcium floods in, and that signals to these vesicles with these neurotransmitters to move towards the membrane and expel those neurotransmitters out. Okay, that is the definition of exocytosis, by the way. Those neurotransmitters can then act on the receptors on the next neuron, the postsynaptic neuron. Keep in mind, this could be your muscle cell as well, right? Remember, acetylcholine from your alpha motor neuron gets released and binds to receptors on the muscle cell. That's what leads to the muscle action potential eventually. Same idea here. It could be a neuron to another neuron, right? It leads to, again, opening of new channels. In this case, chemically gated channels. Why chemically gated? Because neurotransmitters are chemicals. They're not mechanical stimulation or whatever else it could be. They are chemicals. So they can then lead to opening of other ion channels. And now the process repeats, right? You're back to having a graded potential again that can lead to an action potential potentially, right? So back to where we started now. And to stop this process, basically you want to get rid of the neurotransmitters over here. So you can either just bring them back into the axon terminal through a recycling process. You can break them down using enzymes, or you'll see there's actually some spaces here that those neurotransmitters can basically just diffuse away, right? So they won't keep binding to the receptors then. Any questions on any of that? One thing I'll also mention is when we're talking about the hypothalamus and the posterior pituitary, all right, the posterior pituitary released two hormones. Does anyone remember the names of those? They were already brought up twice before. Yep. Right, so oxytocin and ADH. Keep in mind, those hormones are not made in the posterior pituitary. They're just stored there. Okay, they're actually made back in the hypothalamus. That hypothalamus is made up of neurons, right? It's part of the brain, right? So for the neuron to release those hormones, right, ADH or oxytocin, it requires an action potential, right? So everything that we just talked about here is what is happening between the hypothalamus and the posterior pituitary to lead to the release of ADH and oxytocin. Okay, it's the exact same steps because it is a neuron. It's relying on action potentials. Okay, are we good there? I can move on to endocrine next. Okay, so for this, I decided to just focus on hydrophilic versus hydrophobic hormones. I'm debating whether I go into thyroid hormone and cortisol just for the sake of time here, but we'll see. Okay, so again, we can broadly categorize our hormones in two major ways, hydrophilic and hydrophobic hormones. But there are actually four classes of hormones, okay? Protein or peptide hormones, that's number one. Hydrophilic amine hormones, steroid hormones, and hydrophobic amine hormones. Okay, so again, someone earlier brought up the fact of this idea of neurohormone. That is a type of hormone, but it's not a category of hormone, right? So the hormones from the hypothalamus and the posterior pituitary are absolutely neurohormones, right? But they are neuropeptide hormones specifically, okay, because they come from the brain, but they are peptide hormones. They have the characteristics of peptide hormones. So what are the main sort of classifications or distinctions that take place because of this? So hydrophilic hormones, which are your protein and peptide hormones and the hydrophilic amine hormones, are they going to be happy being in the blood? Why? People are saying yes. Why? What's our blood mostly made up of? Water, right? So hydrophilic hormones like water. They're perfectly happy being in that watery environment. Can they pass through the plasma membrane of your cells though? Why? Absolutely, right? So the phospholipids within the cell membrane, right? The lipids themselves are hydrophobic, meaning they act as a barrier for hydrophilic components. So hydrophilic hormones cannot move through the cell membrane. So if you want these hormones to signal to the cells, their receptors have to be outside the cell. Okay, so they're going to be specifically bound to the plasma membrane, sticking on the outside of the cell. Okay, so hormones like ADH, oxytocin, LH, FSH, GnRH, all of those are going to bind to receptors located on the outside of cells. Hydrophobic hormones, like your steroid hormones and hydrophobic amine hormones. So thyroid hormone was the example that we talked about in class. Will these be happy in the blood just normally? Right? They're hydrophobic. They hate water. So instead, they have to be bound to a plasma protein, right? Think about chylomicrons again. When we were talking about lipids, same sort of principle, right? Chylomicrons were those protein shells that allow lipids to be transported in the blood. These plasma proteins are going to basically protect or bind to these hydrophobic hormones and keep them happy within the blood, right? It provides a suitable environment for them to circulate in the blood. But a consequence of that is because they're hydrophobic, they can get into the target cell. They can literally pass right through the plasma membrane. So that means their receptors are actually found within the cell. Okay, they are intracellular receptors because they can actually get into the inside of the cell. One last distinction: protein hormones and hydrophilic amine hormones. We tend to say that they alter pre-existing proteins. So what does that mean specifically? It means the proteins are already made within a cell, and once the hormones bind to the receptor, it causes something to happen to those pre-made proteins. So if we think about ADH, for example, in the renal section, what did ADH do? Remind me. We talked about it before, right? When I was dehydrated, what did ADH do? Yeah, exactly. It's going to reabsorb water in the collecting duct. So what exactly does it do? Well, it's going to bind to receptors on the cells of the collecting duct, and that's going to signal to the collecting duct cells to move aquaporin. Anyone remember aquaporin what channels? Not 1, 2. Aquaporin 2 channels to move into the luminal membrane of the collecting duct, right? Those channels are already made. They are pre-existing proteins. ADH binding to its receptor just moves those proteins from the inside of the cell to the cell membrane. And now you can absorb water into the cell and eventually back into the blood then. Compare that to your hydrophobic hormones, which are binding to intracellular receptors. They tend to affect the production of new proteins, right? So the thing is, because they're intracellular receptors, what ends up happening is the hormone and the receptor, when they bind, they'll physically move into the nucleus of the cell and lead to the literal transcription and translation of new proteins, right? So if you think about cortisol, for example, right? Cortisol was a steroid hormone. It does all sorts of things. It acts on the liver to promote gluconeogenesis, for example. It's going to go to the liver and it's going to promote the production of genes that leads to gluconeogenesis. Now, which of these two do you think will take longer? Making brand new proteins from scratch or changing pre-existing proteins? Right, it's going to take longer to make new proteins, right? So I can't remember if Dr. Bell made the example or not, but I like this example. It's the equivalent of going to the grocery store and buying a pre-made cake versus making a cake by yourself. It takes way longer to make a cake from scratch than it does to make a pre-existing cake, right? So that is how you can kind of rationalize that. So hydrophobic hormones tend to have longer time of onset of action compared to your hydrophilic hormones, which tend to be a lot shorter. And again, that's because of the fact that hydrophobic hormones have to make brand new proteins. They can actually get to the DNA and promote the production of new proteins. Okay, I'm going to give you guys a choice. We can move on to sensory because we only have around 20 minutes left here. We can move on to sensory, or we can talk about thyroid hormone and cortisol. Sensory? We'll do show of hands. Sensory? Thyroid hormone, cortisol, any of that nonsense? Here, we got one. Sorry, dude. The PowerPoint slides are uploaded on OWL, or I will upload them on OWL so that you can always have access to them. Okay, so when it came to sensory, I basically decided to go with two topics. Receptive fields and the transduction of vision and light. Okay, so I've already kind of briefly talked about like sound already. And again, my tutorial slides go into a lot more details on how sound is perceived. I don't think that's a very complex thing. I think vision tends to be a little bit more of one of the more complex things. So we'll start with this, and then we can talk briefly about receptive fields. Because if I had to guess, I would totally expect Dr. Everling to ask something on receptive fields, especially with touch, for example. He might ask something about pain too. That's another thing he could ask. I'm not going to belabor this from start to finish like I did in tutorials. Again, my PowerPoint slides go into details. All I'm going to say is your left and your right eye both contribute to the left visual field and your right visual field. So what does that mean again? If I were to stand directly in the middle of this class, everything directly in the middle of me is my center. Everything to the left of that is my left visual field. Everything to the right of that is my right visual field. Both eyes contribute to both of those things. When it comes to what information is reaching your visual cortex, your right visual cortex is seeing your left visual field. Okay? Your left visual cortex is seeing your right visual field. The bottom line is because both eyes are contributing to both visual fields, that means that information from both eyes, at some point, something has to cross over to get to the correct side of the brain, to get to the right visual cortex, or the correct visual cortex, I should say. Right? So for example, your right eye, if it's contributing information to your right visual field, for example, right? So you can see this part over here, the nasal retina, is contributing to the right visual field. Well, if it's contributing to the right visual field, that means it has to get to the left visual cortex, right? Again, everything is kind of flipped by that way. So something has to happen. You have crossing over taking place at that optic chiasma for that to take place. Okay? That's the gist of what I want to explain. In terms of what I can expect some questions from Dr. Everling, he might say, which of the following lesions would contribute to losing your whole right visual field? Okay, so the whole right side of your vision, right? So everything on this side. It's not going to just be your right eye, right? It's going to be everything on this half of the brain past the optic chiasma, right? So your left optic tract, as it's called, left lateral geniculate nucleus, left optic radiation, or left visual cortex. Any of that will lose the right visual field. Is that all good? Sort of, kind of, ish? Maybe? I'm going to take that as a yes, because I don't want to explain that again. Anything else that he could ask? He might ask, what is the consequence of severing one of the optic nerves? Okay, so keep in mind, optic nerve is literally the information coming from the eyes, right? So losing your right optic nerve is the equivalent of losing your right eye. What do you lose? Basically information that just the right eye sees, right? So it's not going to be your right visual field, because guess what? Both the left and the right eye contribute to that right visual field. And again, you can see this, right? If you were to cover your right eye, you could still see things on the right half of that visual field, right? So both eyes are contributing to both sides there. What you do lose by losing your right eye is basically the peripheral vision of the right eye, right? Everything that the left eye cannot see, that's way over here, right? My left eye can't see way over here. That's what gets lost by losing your optic nerves. Okay, so loss of your optic nerves, basically you're losing that kind of like the ends, like the periphery of the vision. Loss of your optic chiasm means you lose the crossing over events. Well, what do you see in your crossing over events? It's basically the right periphery and your left periphery, right? Anything that had to cross over to get to the correct side of the visual cortex, right? So if you had a vertical lesion of your optic chiasma, you'd basically be left with something like this, right? You're basically what both eyes see, which is the binocular vision, excluding the periphery of both the left and the right eye, because that's what has to get crossed over. Okay, any questions on that? Okay, let me pull up another PowerPoint here. Okay, yeah, receptive fields. Okay, so if I had to guess what Dr. Everling could ask on the exam, one thing he could ask is what, actually, that's a little bit too detailed, I'd say. I wouldn't worry about that. Well, I will say it anyways. One thing that he could ask is what sense does not have to pass through the thalamus. As you might have remembered, maybe, that pretty much every single sense, like taste, touch, vision, hearing, and balance, has to get through the thalamus. The one exception to that is smell. Okay, so smell coming from your nose literally goes directly to your the part of the brain that processes that information, the olfactory cortex and olfactory bulb. Okay, I don't think he would ask that because that's a very, very like minor detail thing, but that's just something to keep in the back of your head. I don't think he'll ask anything on that or that, I hope. Probably nothing on that. Again, I'd say the main thing from the very first lecture that he talked about is kind of the distinction between complex neural receptors and special senses receptors, and receptive fields, probably the other thing. Okay, so I'll ask you, complex neural receptors, how do they differ from special senses receptors? And don't overcomplicate it. This may be like, oh man, I don't remember this stuff, but look at the names of what we're talking about here. What are neural receptors versus senses receptors, right? The difference there is the word neural, right? It says complex neural receptors are neurons, right? They have neurons, literally part of them, meaning they can generate action potentials. That is the big distinction. Special senses receptors are not neurons. They are completely separate cells that have to signal to neurons. Okay, so they are separate cells from the neurons. They cannot generate action potentials. So what can they generate then if they can't generate action potentials? Any thoughts? What was the other thing that we talked about? Not action potentials, but graded potentials, right? Basically. Now, in this case, he gave them a different name, receptor potentials, as he called them. Right over here, local current flow, receptor potentials. But don't confuse that. They are graded potentials, right? They are going to have either mechanically or chemically gated channels that are going to get activated. And if the stimulus is strong enough, they're going to release a lot of neurotransmitters that's going to act on a neuron. And if that hits threshold, you have that action potential taking place then, right? So keep those things in mind. Don't overcomplicate stuff like that, right? Look at the way it's called. Complex neural receptors are neurons. They can make action potentials. Special senses receptors are just receptors that detect sense, right? Whether it's vision or what was it? It's not touch and it's not smell, right? So it's vision, hearing, balance, and taste specifically where you find these special senses receptors. Okay, so they can generate receptor potentials or graded potentials. They do not generate action potentials. They signal to a neuron to lead to action potentials. Yep. Like, where would you find these? Like the complex neural receptors, you mean?
No, like where would you find, because you said we're only getting vision, hearing, balance, and taste, like where would you get like touch and
Oh, touch is part of somatic senses. Smell is part of olfaction. Okay, so those would be involving complex neural receptors. Now that is kind of important, but I don't think Dr. Everling would ask that because that's what I would consider as a small detail for the final exam. Again, please don't like come after me after saying that and he asks that question. Like, study everything. I, as your TA, can only answer what I can. I don't see the exam and I don't participate at all in making it. This is just my thoughts on it, right? So just keep those things in mind. Receptive fields. Again, don't complicate this. If something is touching my arm, that means a neuron is getting activated. But if a neuron is located over here, is that neuron going to get activated if something is touching me over here? No, that is what a receptive field is. Okay, so the space that a stimulus has to fall into to activate that neuron, right? So same with vision. If I'm looking directly at this chair, okay, that's a bad example. If I'm looking at the lights, those photons are literally coming into my eyeballs and they're activating receptors specifically where those receptors are looking at that light. They're not going to activate receptors that are way, way over here, for example, because they're way out in the periphery, right? So those stimuli, whether it's touch or sound or taste or vision, those stimuli, those molecules have to fall within this area to activate that neuron. And we call that area the receptive field. Okay, so I can see Dr. Everling asking a question on receptive fields on your fingers versus your back, for example. Right, so what did we say was true about the receptive fields on your fingers compared to your back with regards to the size and the number of them? Any thoughts? Yep.
We like more on the.
Exactly, right? You have way more and smaller receptive fields, which is why you can very easily differentiate things when you're touching them. Like you can feel the texture very easily. There's a lot more information and a lot more resolution to be able to make those distinctions. I don't interact with the world with my back. I don't know about you, but that because of that, I'm not going to have as much receptive fields or as many receptive fields in my back. I'll have way bigger ones so that I can still feel those things, but I can't tell between whether this is two things touching me or one thing touching me because they're both falling within one receptive field. And it's going to activate one neuron. Okay, so I sense that or I perceive that as one thing touching me. My fingers have a way better idea of differentiating between those things because there are way more receptive fields there. Does that kind of make sense? And hopefully that makes sense from an evolutionary perspective, right? Again, I'm interacting with the world with my fingers, not my back. Okay, next up is motor. Everyone's favorite unit, probably, maybe. So basically what I chose to do for motor is the basal ganglia and Parkinson's. Okay, so again, the other major aspect of motor was your alpha gamma coactivation with your muscle spindles. And a third one was your cerebellar pathways. Okay, so the idea of cerebellum. Again, we don't have too much time left here. We have 9 minutes. So this is what I've chosen to focus on. But I could see Dr. Everling asking a question on muscle spindles, right? What happens when you passively stretch your arm compared to an active contraction? Go back to my PowerPoint slides if you have to, because I go into details on those things, right? But specific neurons, remember alpha motor neurons or gamma motor neurons activating specific things in your 1A afferent fibers and your 1B afferent fibers. Remember all that? Study those things because you can see a question potentially on that. Okay, so let me see if I can find the PowerPoint slide for motor here. Okay, so one thing I'm just going to say for the basal ganglia and Parkinson's, again, I'm not going to go through all of it again because it's going to take like 10, 15 minutes on itself. If there's a question on the exam on Parkinson's, I could see the answers being something along the lines of what causes it. And basically the major cause of Parkinson's is this right over here, the substantia nigra pars compacta being lost and losing dopamine. So again, if you were to work through all of these pathways, what you would find is in Parkinson's, by losing dopamine, the other big key takeaway from all of this is that this structure right over here, the globus pallidus internal segment, its activity increases. That is the key takeaway for Parkinson's. The activity of this increases because you've lost dopamine. And again, you can work through the pathways, go through all this, the excitatory and inhibitory and all that nonsense. The point is, this structure here, the activity increases. What is the consequence of that? It inhibits the thalamus and prevents it from exciting the motor cortex. So what does that mean? It means that you're inhibiting the structure that's going to promote the activation of the brain area that's going to allow you to move, right? So you lose excitation to the motor cortex. What do you think your movement is going to be like if your motor cortex is less excited? It's going to be pretty poor, right? You're going to have slow, rigid movements. Look at the symptoms of Parkinson's. Slow movement, rigidity, slow movement initiation, hypokinesia, slow shuffling steps, because the motor cortex is not being activated to the degree that it needs to. Okay, so again, if there's a question on Parkinson's, loss of dopamine because the substantia nigra pars compacta is lost, globus pallidus internal segment activity is increased, motor cortex activity is decreased. Those are the 3 major things in that diagram that I would remember for the exam. And God, I have no idea what to say about the cerebellum. I can't even fathom what a question would be like on that. So, no, I got nothing. Yeah, good luck with that. Sorry. Study it. Just look it over. Again, it's a multiple choice exam, so maybe something can be triggered in your brain the day of, and or he might not even ask a question on it because he knows it is a tricky concept, especially for a final exam. So take it like that. Okay, we have 5 minutes, and we still have cardio, renal, and respiratory to go through. And again, we don't have time to go through everything, so I'll leave it up to any of you to kind of decide here. What do we want to finish up with here? Anything you'd like. Or any other topic at all. Or nothing. Yep. Muscle spindles? Okay, sure. Okay. So on the left side of the diagram is your muscle, right? This could be any muscle in your body. It could be your triceps or biceps or whatever. The point of the muscle spindle is that it is basically the receptor detecting the stretch of the muscle. Okay, so you literally have neurons that are wrapped around part of the muscle, and I'll say it from now, it's the inner part of the muscle. Remember the intrafusal fibers, that if the muscle is being stretched or if it's being contracted, these neurons can detect the length of that muscle. So the whole point of the muscle spindle is to detect the length of the muscle. Okay, so we're going to talk about the sensory aspect in just a second, but let's talk about how these muscles work. Keep in mind, for a muscle to contract, you need motor neuron activation. So you have to have an action potential on a motor neuron that's going to then activate the muscle cell. Right, so as you're studying, think about all those steps that are involved there, right? So acetylcholine binds to receptors, leads to the end plate potential, leads to an action potential, leads to it going down the T tubule and all that stuff, right? To make those connections as you're studying for these things. Okay, so the muscle fibers that we see sort of on the outer part of the muscle is what we refer to as the extrafusal fibers. If I'm lifting something like my bottle of water right now, these are the fibers that are contracting. This is not very heavy, but if I was lifting something heavy, these are the fibers that would be contracted to allow me to actually lift that object. Inside the muscle are intrafusal fibers. They don't play as big a role in contraction and force generation, but they play an important role in muscle length detection, which we'll see here in just a second. Okay, so alpha motor neurons are important for allowing our muscle to contract, to give us our contractile force to lift things. We have another set of neurons, gamma motor neurons, that activate the inside fibers, the intrafusal fibers. Okay, so when these activate, same idea, they can release acetylcholine and they lead to the contraction of intrafusal fibers. Now, these again don't play as big a role in force generation. They play another role in making sure that information is continuing to be sent to the brain on the length of a muscle. So let's see what that looks like here. Okay, I'm going to skip over Golgi tendon organ. I will talk about this though. So I told you we have to have neurons wrapped around the muscle for you to detect stretch, right? Sensory neurons. We call those 1A afferent sensory fibers. They are the ones that are basically wrapped around the intrafusal fibers and can detect the length of that muscle. I'll talk about that. So now we can talk about this. Okay, so if a muscle is being stretched, what is happening to its length? Is it increasing or decreasing? It's increasing, right? That's what stretch means by definition. So what happens to the muscle spindle? Well, the length is increasing. Therefore, the activity of that receptor, those 1A afferent neurons, is going to go up, right? Because it's detecting length. If the length is increasing, its activity is going to increase. Okay? I'll mention here the Golgi tendon organ too, because it makes a lot of sense. This is found at the tendon, right? The connection between your muscle and your bone. If you're stretching your muscle, you are creating tension on your bone. So you'd expect the Golgi tendon organ, which is detecting the tension here, to also increase in its activity because there's more tension being generated there. Not important. Let's get back to the muscle spindle. Now, if you were to contract your muscle, think about the sliding filament theory. What's happening to the sarcomere? It's shrinking, right? It's compressing inward. Remember those Z lines getting closer together? They're coming together. The muscle is shortening. That is the definition of muscle contraction. The muscle is shortening. What's happening to its length? It's decreasing, right? So because of that, if the muscle spindle is involved in detecting length and the length is now decreased, its activity is going to go down. Here's the problem with that. This is, by the way, not what happens normally, but this is what would happen if not for this idea of alpha gamma coactivation, which we'll talk about here. But this is what would happen. Here's the problem with that. Your 1A afferent fibers are sending information to your brain about the stretch of your muscle, the length of the muscle. If you were to contract it far enough that the length were to decrease enough, it will eventually stop firing. Because again, remember, it's detecting length. If the length is decreasing, it's going to reduce its firing rate, right? The muscle spindle is getting less and less activated. Eventually, you get to a point where the 1A afferent fiber is no longer sending information to the brain. Is that a good thing ever for your brain to stop receiving information on your limbs? No. So you do not have this happening. This is terrible. Instead, we have something happening called alpha gamma coactivation, right? So alpha motor neurons activate. That's what led to the contraction of the muscle in the first place, the active contraction, right? That's what leads to the muscle shortening. That's specifically of the outside fibers, those extrafusal fibers. Then you have gamma motor activation, which is going to cause the contraction of the intrafusal fibers. So what exactly happens? It's going to cause them to basically contract and get pulled tightly again. So in this example, if the whole muscle is shortening, what happens to the inner fibers? It's basically going to go like this. Okay, because the inner fibers, without alpha gamma coactivation, inner fibers are not having any signal taking place to them. Once the whole muscle shortens, the fibers basically do something like this. Okay, they become limp, and there's no length to this. This is why you're not sending any information to that 1A afferent fiber anymore. No information is going to the brain because there's no length here. But with gamma motor activation, you cause those muscles to basically contract again. They become more taut, and now there is length to this. Right? This can be detected by that 1A afferent fiber. And that is what allows you to continue to signal to the brain what is happening to the muscle length and the stretch of the muscle. Does that kind of make sense? Sort of? A little bit? Are you sure? You're allowed to say no. Okay, perfect. Just checking. Okay. It is 101. Again, I didn't get through cardio, respiratory, or renal, but I think reach out to by email or discussion forums if you have to. I'm happy to meet and answer any questions. Otherwise, good luck with the exam. Unfortunately, I will not be there for this exam, but I know you guys are going to do great, and I wish you all the best of luck with your undergrad degree. If you see me around campus, feel free to say hi to me. No pressure, though. Thank you.